Okay, let's start. Welcome all to the second day of the first international symposium on continuous real-time biomolecular monitoring, biomolecular sensing. My name is Sven Ingebrandt. I'm from RWTH Aachen University, and I'm also one PI of one of the competing teams here. Today we will hear in the afternoon a session about SPR and SERS biosensing. And I'm happy to introduce the first speaker. We have a few audience here in the room, but I got to know that there's quite a crowd in the online audience. We want to hear talks of 15 minutes plus 10 minutes discussion, so please stick on time. And I will take the questions here first from the audience and then also from the online audience. So I'd like to introduce the first speaker. The name is Daniel Aili, and he is from Linköping University in Sweden. And Daniel is a professor in the Division of Biophysics and Bioengineering at Linköping University, and he is professor of molecular materials. Just a brief summary of his CV. He did his PhD and first postdoc with Molly Stevens, who is very well known in the field of biosensors. And then he went to the Imperial College London, and after another stop as a postdoc in Nanyang University in Singapore, he went back to Linköping and is now heading the group of molecular materials. So we are looking forward to your presentation. Please start.
Thank you very much for the kind invitation and for the introduction. So I'm happy to be here to talk to you about real-time nanoplasmonic biosensor for detection and quantification of monoclonal antibodies in bioproduction. Can you hear me?
No. Now we hear you. Now we hear you.
Okay, sorry for that. Then we take it from start. So thanks for the kind invitation and introduction. So I'm really happy to be here to talk to you about real-time nanoplasmonic biosensors for detection and quantification of monoclonal antibodies in bioproduction. So, short disclaimer, I'm a co-founder, shareholder, and chairman of the board of Aarhus, which is a startup company providing LSPR-based fiber optical biosensors for the biopharma industry. So, what is a biological drug? So biologics for shorts are medicines produced using living organisms such as bacteria, yeast, or mammalian cells, rather than being synthesized from chemicals as most conventional medicines. So these are typically large, very complex molecules such as proteins, antibodies, or nucleic acids. And these medicines are very complicated to produce and are sensitive to the manufacturing processes. And this also makes them extremely expensive to produce, resulting in very expensive drugs that are not affordable to a large population or large fraction of the global population, unfortunately. Despite of this, these are blockbuster medicines. This is just the 10 highest valued drugs currently available. And as you can see, 8 out of these 10 are biological drugs, and the revenues for these drugs are, numbers that we can barely imagine. But they provide treatments for diseases that are, really providing revolutionizing new treatments. So highly valuable. Out of these 10, 6 are monoclonal antibodies. So this is really a large and very important group of biologics that we are currently targeting our sensors for. And as I mentioned, the production of biologics is very complex. And that's what a typical production process looks like schematically. So discrete production steps, an upstream process where you culture your cells, producing the product, and then a number of downstream steps where you purify these. And between each, or even within each production step or process step, you have to retrieve samples, send them off for quality control and analysis to make sure that you have the right product of the right quality and have been able to remove all the undesired byproducts or contaminants. So this makes the whole process very tedious. It's a process that happens in the dark, so to speak. What the industry is striving for is to essentially automate several production steps to be able to connect them and intensify the process. In order to do this, you need real-time information on, for example, product titer, presence of product variants, and contaminants, because this information allows you essentially to control the process in real time, which can provide significant improvements in quality of the of your products, result in smaller factory footprints, smaller environmental footprints, and lower cost of the drugs. And this is also the aim of our work, to essentially facilitate this modernization of by the biopharma industry. So we develop LSPR, so local surface plasma resonance-based biosensor systems for rapid inline bioprocess monitoring to facilitate production of biopharmaceuticals. So we have these devices, or developed these devices, and a key component of these devices is this little sensor chip where we have gold nanostructures. So this is the key of this technology. And gold nanoparticle, when you irradiate it with light, you can induce collective oscillations of free electrons in the metal nanostructures, which results in a very pronounced absorption spectra of the light. And this is what it can look like, the green curve here. And the position of this plasma band is dependent on the refractive index or the dielectric constant of the surrounding media, around 20 nanometer from the nanoparticle surface. So when we change the local environment here by, for example, absorbing a biomolecule on the surface, we also shift the plasma band. And this shift, the magnitude of that shift is dependent on the change in refractive index, and thus the amount of biomolecules on the surface of the nanoparticles. So we mobilize these nanoparticles on the sensor chip. The sensor chip is mounted in a flow cell that then goes into the stream of the process. So this is just one example where we have mounted the flow cell on a R&D or process development chromatography system. And the readout is done using fiber optics. And a key element here, so these measurements are done in reflectance mode. We have the nanoparticles on the surface here, but irrespectively, a key part for this to work is a robust surface chemistry. So we functionalize the gold nanoparticle in the sensor chip in order to provide unspecific binding while allowing immobilization of suitable ligands for binding over analytes. And we monitor the shift of the LSPR band in real time to obtain a sensogram. Those of you who are familiar with BIACOR or SPR measurements recognize what this looks like. So we see the binding of our analyte in real time and the dissociation. And when looking at the sample from a bioprocess, it can look like this. So this is, you recognize the binding of the analyte, in this case IgG, which is the most common monoclonal antibody used, at different concentrations. So essentially the amplitude of this shift is dependent on the concentration. And in this case, we use protein A as a ligand, which is a high affinity ligand for IgG. There is virtually no dissociation under these time frames here, but you can peel off the analytes by reducing pH, essentially do a regeneration, and this allows you to go back to baseline, introduce another analyte, in this case same concentration of the same antibody or sample here, showing that we can regenerate and reuse the sensor chip without deteriorating the sensor response. So the sensor chips are quite robust, but as I mentioned also, prevention of unspecific binding is a key here. And this is because the sample matrix often in this context is extremely complex. So in addition to, at least for the upstream samples, a lot of cells, so many millions of cells per milliliter, it looks like a thick soup essentially, and plenty of different proteins there. So the sensor chips need to be able to withstand unspecific binding under these extreme conditions, while also providing specificity and minimize matrix effects in general. And this is what it can look like in these kind of samples. Here we have two samples spiked with known concentrations of monoclonal antibodies. And from a calibration curve, we can determine the exact concentration here. And the precision in these measurements is independent on the presence of cells. So we can essentially get the information on titer very quickly. This is still not in line in the process, but at line, but 2 minutes compared to current methods for the analysis, which typically counts in hours and with very low sample volumes. But in order to turn this into an inline methods, we will need inline regeneration of the sensor chip also. And here you can see again the sensor system mounted in the chromatography system. And here the purpose is to be able to monitor what leaks out through a affinity chromatography column during the loading of the column. And here you ideally don't want to overload your columns because that results in a loss of extremely expensive products. But this can happen, and with the conventional sensors, you can typically not see this because of the complex sample matrices. So with the UV sensor, there is essentially just no signal at all coming out when we, with this LSPR sensor, can clearly see that you have a product breakthrough, you're losing sample. And in a normal case, you would abort the loading and start washing and eluting your sample here. And the nice aspect about this, which allow us to keep this as an inline sensor, is that the sensor surface is regenerated as you wash out your column, and it's ready to go again for the next sample. And we can see when we zoom in here, the clear binding curve from the LSPR, whereas the UV sensor responds slower at a much later time point and with a lower signal. And we can also directly from the derivative and the slope of this binding curve determine what concentrations of IgGs are being lost during the overloading procedures. Also, so we can keep track of titer, we can keep track of how and when to optimize your chromatographies during conventional purification, but we can also use this technology to detect aggregates. And aggregates is a huge problem because these are undesired byproducts that naturally form during the production process and must ideally be removed or at least kept constant in your drug product. So these must be monitored. And we hypothesized that dimers of and trimers and higher order aggregates would bind with higher avidity to our sensor surface, since it essentially can bind to multiple ligands on the sensor surface. So a system, or essentially a situation like this. And this turned out to be true, not necessarily at pH 7. Then we cannot see any differences in aggregate and monomer concentration. And that's because we have, using a protein A sensor chip, no dissociation of our analytes. But when reducing the pH, we start to see a more rapid dissociation of monomers compared to aggregates. So from the dissociation rate constant, we can determine the presence and concentration of aggregates. And this we can also do in real time during affinity chromatography, enabling fractioning and removal of aggregates already in the affinity chromatography step. So to conclude, we've developed an LSPR biosensor system for real-time inline bioprocess monitoring. It's very sensitive and it provides reproducible data on primarily IgG, which is currently optimized for, but biologics in general. And the sensor can provide real-time information on product concentration and aggregate content, and also sensitive detection of product breakthrough in chromatography. So I would like to thank my team members and, of course, collaborators and funding agencies. And thank you.
Yeah, so thank you very much, Daniel, and also thank you for sticking perfectly in time. So we have now 10 minutes of questions. I would take questions here from the audience. Any question here? Jean-Francois?
Thank you very much for the presentation. So my question regards the surface chemistry that you briefly mentioned. So can you give a bit more detail on what surface chemistry actually prevents fouling from the cells?
Yeah, so that's essentially multiple layers of self-ascending monolayers on the nanoparticles and on the steel surface here. I can't unfortunately go into all the details since some are protected for commercial purposes. But polyethylene glycol is an important component here, and it's been optimized in over multiple years essentially, because this indeed is extremely challenging to prevent the fouling essentially, and it's extremely robust.
And if I can squeeze a second question, actually, so I would like to see if you have any means to monitor the quality or the affinity of the antibodies coming out. So you measure concentration, but can you measure their activity against the target they will be intended for?
Yeah, absolutely. I mean, in principle that's possible. I mean, this is a generic sensor technology. So if you have use your essentially antibody target as the ligand on the sensor surface, then you can retrieve all the information on the affinities and kinetics of binding. So that's definitely possible.
Okay, thank you. Any more questions from here?
I had a question about your the final part. So where you try to distinguish aggregates from monomers. I can imagine that's quite tricky, because aggregation is concentration dependent. You also showed that at pH 7 there's no dissociation of the monomer at all. So yeah, so to what extent is this industrially relevant? Because if things start to aggregate at high concentration, it's just a concentration effect, basically. Or are there examples where if antibodies are produced, they're not homogeneous and certain forms have a higher tendency to aggregate? Can you elaborate a little bit on that? And the other question I had, I think the detection is based on a multivalent interaction, which means that it's not only dependent on the aggregate, but also on your sensor surface, so the density of that. Is that something that you play with in order to specifically detect aggregates versus monomers?
Yeah, very, very relevant questions here. And the industrial relevance, I would say, as far as collaborators in the industry, they indicate large interest in this, and because this is something that they struggle with. For example, upstream in the actual cell culture, you want to run the process. I mean, typically you have a set time, but during at least the process development stage, optimize the conditions to avoid aggregation of your products to the largest extent. And then having like an online sensor where you actually can, as we did in this example here. So this is a simulated online condition. Essentially, you retrieve samples. It doesn't go back to the bioreactor. So it goes down to waste, and then you can essentially adjust the pH and look at the dissociation constant, and you can get this information without essentially sending off sample to a quality control lab. So that's one purpose, but also when we can do this inline during affinity chromatography, as we showed in this work, then you can essentially, it allows you to essentially reduce the or optimize the purification step of these antibodies, potentially skipping an entire chromatography unit operation by being able to separate and identify your aggregates during the affinity chromatography step, which is currently not possible, because if you look at the conventional sensors that are available, we don't see any signal, or at least a minute signal, that is hard to regulate anything on from the conventional sensors. Here we have a huge difference in sensor signal for relatively small difference in aggregate content. So when you start seeing this slower dissociation, then you know that you have a large amount of aggregates coming out there. And I didn't have space for including that, but we can combine this with a pH elution gradient in order to also enable separation of aggregates during this step, while also enabling then the detection of aggregates. So that can facilitate process development and actual production.
Okay, thank you. So we have another live question here, please.
So hi, my name is Vivek Pashori, so from RWTH Aachen. So this discussion about the aggregates, yeah, so this also I find very interesting. I was wondering here, so about the amount that you see, yeah, so in the real-time characteristics, yeah, so you can distinguish that, okay, we have different amount of aggregates, but I wonder how the size is relevant, and if you employ some complementary characterizations, yeah, so to see the size distribution of these aggregates. Because in this plasmonic approach, I guess the size also would matter a lot. Also, what you see as a response.
Yeah, in principle, it could. I mean, larger molecules or higher molecular weight should give you a larger response. So from that perspective, that's correct. But here, I also believe, I mean, since the surface density of the bound molecules also influences, it's not the direct or simple relationship here. And since also aggregates take more space on the sensor surface. So we haven't really been able to correlate this to size in any way. We also don't know how these aggregates orient on the surface because we have a quite limited sensing volume with LSPR. It's roughly just 20 nanometer from the surface, and part of that is occupied by the surface chemistry and the ligand. So it's roughly 5 nanometer or 10 to 15 nanometer of, I mean, space where we can detect things. So a large aggregate, part of it will be outside the sensing volume, we suspect. But this is definitely something that it would be very interesting to look more into. Because, yeah, and also when it comes to detection of other compounds of varying size, potentially enabling some sort of multiplexing. But that's yet to be researched. I forgot to answer the question regarding the ligand density of the surface. And yeah, that's in principle matters, but we have not, at least not with protein A, been able to space them out sufficiently to avoid completely avidity effects. So that's actually one of the outstanding problems, because sometimes you don't want to see these aggregates, you just want to measure your monomers.
Okay, thank you for your detailed response. Yeah, so I appreciate it.
Okay, also thank you from my side. So are there online questions? So online audience, if you have questions, please type in the chat, I guess. So if not, so we have 1 minute left. I will ask a fast question, and I ask you for a quick answer. So for me, it was not completely clear in which format this sensor is brought into the media. So was this a flow cell, or is this a dip-in, or how is this produced, or how is this practically done?
Yes, that's a very good question. So it's a flow cell. If you can see from this image here, we have the flow cell here, and we're docking in the sensor chip here. So this is essentially connected to the process flow.
Okay.
And then there is a fiber optical connection to the actual optical unit where we have light source and detector.
Because you mentioned you are very sensitive to the interface, yeah? So you need to bring the molecules there in a decent time.
Yes.
Yeah.
But they go through the flow cell. We are also developing a dip-in sensor, what a probe that can be mounted in a bioreactor. But that's yet to be tested live.
Okay, so I think we are done with the discussion, so perfectly in time. And also thanks again, and thank you for stepping in as a replacement for the first talk. So
thank you.
Thanks.
Thank you.
So I hope that now the connection to the second speaker is working. Weiqiang? Yes.
Hello, can you hear me?
Perfect. We hear you.
Okay, great.
So let me introduce you. So Weiqiang Zheng, he is actually in New York at the moment. He's professor at New York University, and he is a professor in the departments of biomedical engineering and mechanical and aerospace engineering. So two affiliations somehow at New York University. He earned his PhD degree in mechanical engineering at Michigan, and I don't want to read down all the awards. He won several awards, and he works on lab-on-chip and organ-on-chip, and we hear something about single-cell cytosine secretion and detection, together with CAR T-cell therapy. So this is quite interesting topic. So please.
Okay, thank you for the introduction, and it's great opportunity for me to discuss our research on this new nanoplasmonic sensor for real-time monitoring single-cell cytokine secretion, which we applied for cancer CAR T-cell immunotherapy. Let me see. So short background here. We all know that the cancer treatment is always a challenging task. So in recent years, the cancer immunotherapy, people start to use patients' own immune cells like T cells to kill their cancer cells. So the CAR T immunotherapy, for example, they will be engineered using the patient's own T cell, and then they can recognize the antigen like CD19 on the B cell for leukemia, and then they can kill the cancer cells. During the treatment process, the cytokine secretion signature is an important marker to indicate the immune response and clinical efficiency. For example, here you can see that in our study we use this TNF-alpha, PFN, and alpha gamma to as a marker to indicate CAR T cell activation and the cytotoxic function. So we see that like a patient CAR T cells have a weaker immune responses and functions, and also we test different CAR T cell designs, different CAR T cell products. We can also see different. I'm not going to look into details here about what's the advantages different designs, but you can see that their cytokine secretion, like TNF-alpha, alpha gamma, is a marker which showing like IL-18 CAR T cells are really more efficient than the other designs. So this is one major application of the cytokine detection to identify the CAR T cell efficiency, but also we know that the CAR T cell went through the killing process, and they have to happen in the tumor microenvironments, and all the immune cells, suppressive cells in the tumor microenvironments will interact with the CAR T cells and tumor cells. And during this process, you can see different cytokines are activated and secreted in dynamic way, and not only the key cytokines that's as a marker for the CAR T cell activation and function, but you can see that there are a lot of cytokines are important in the effect function, stimulation, chemoattractive, other functions during therapy. And the third aspect for cytokine detection for the CAR-T therapy is the cytokine storm. So the severe cytokine secretion will cause strong inflammation. So these kind of cytokines are secreted by the different cells, and they should be monitored in a time dynamic way. And especially we want to know the mechanisms how cytokine storm initiated, which is not really clear now. We only know like IL-6, the major marker, but what's a signaling network causes cytokine are unclear. So our approach using the LSPR, which is similar to Daniel's approach. So we can use this in golden nanoparticles, right? We functionalize it with antibodies to target different cytokines, for example, the IL-6, TNF-alpha, TGF-beta. So the cytokine binding to the nanoparticles will cause not only the frequency shift, wavelength shift, but also the intensity increase of the exciting light. So instead of using frequency detection, which might be more accurate, but we choose using the intensity measurement. We use a filter out the wavelengths of the peak, and then we measure the intensity increase by using the dark field imaging. So the advantage of this way is you can do an array of the sensors so that you can do multiplexed measurement for multiple cytokines. And the LSPR, the advantage for that is you don't need a secondary antibody to labeling, so that means we can use it for label-free real-time sensing for different cytokines. Like here, you can see this kind of calibration curve, but also showing that we can measure cytokines continuously. And a simple example, just we apply this LSPR sensor to detect different cytokines from the tumor-associated macrophage, and we just showing the differences among these different conditions with multiple cytokine detections using our plasmonic sensor. But our new device not only use single golden nanoparticles, so we instead of using one particle functionalized with antibody, so with a pair, like two particles linked with a DNA aptamer. So, and in this case, when the DNA aptamer, which is designed to specifically capture similar to the antibody, but they have much smaller molecule size, so they can capture specific cytokine like IL-6 molecule. So whenever they capture the one IL-6 molecule, for example, so they will bend and shorten the distance between the two nanoparticles, so enhance the plasmonic coupling and with the enhanced intensity of this dark field in the dark field imaging. So you can see that we can have a lot of a pair of golden nanoparticles, which we call nanoplasmolurals, biosensor. And you can see here it's a real T cell, and that's surrounding the T cell. We functionalize a lot of nanoplasma rulers on the glass substrates, and you can visualize how the cytokine secreted from the single T cells. So we can simply just capture a lot of dark field images, and then we can do real-time monitoring of the cytokine secretion events. So the basic concept, as mentioned, we just link the two golden nanoparticles with the DNA aptamer, and then the DNA aptamer are designed specifically, to capture specific cytokines, and we also doing some simulation prove that indeed if you shorten the distance, the plasmonic intensity, light intensity will be further enhanced compared to single nanoparticles. So we did some calibration with our system. For example, we loaded a different concentration of cytokines into the like microwell with the sensors functionalized with the glass substrates, and you can see that we can monitor the cytokine, we can measure cytokine concentrations using our sensor. And our sensor have a limit of detection about 60 picogram per ml for concentration measurement, and we have a very good temporal resolution and spatial resolution because these golden nanoparticles are very small, and we can do 10 sequence imaging, and then can measure cytokine secretion levels from the single cell. And here we're just showing the examples how the top row showing this cytokine, the image quantified from the dark field image, and the concentration increased over the time. And then multiple cells have different, like say, different secretion profiles, not only the concentration but also the spatial distribution. You can see they are quite different. And next, we try to study how the CAR T cells or T cells are interacted with leukemia cells. So you can see here, there's two cells, like one is a CAR T cell, another is B cell leukemia cells. And you can see the cytokine secretions are more enhanced when here we have two cells, or instead only have a monocyte or only T cells or the tumor cells. They don't have too much secretion. So that indicates that our sensor measurement indicates that the cell-cell interaction indeed will be critical for the cytokine communication. And we did more detailed studies. Like we can see that we look into different single-cell cytokine secretion profiles. We can see that some cells have an isotropic cell cytokine secretion profiles. Some that have anisotropic secretion profiles. So there's more details we need to look into that. But we just found these differences among different single cells. And lastly, we just to study the three cell clusters, like CAR T cells, tumor cells, and also macrophages, because we know that the macrophage is a source of the IL-6 during the cytokine storm, and they are activated to secrete. The macrophage activated by the CAR T cells during this CAR T cell treatment. So you can see that in the three cell groups we have a enhanced alpha gamma and IL-6, and because we can monitor single cell secretion profiles and patterns, so we can see that alpha gamma is produced mainly by the activated CAR T cells, and IL-6 is produced in response to the CAR T cell activation and recognition of leukemia cells. And just more, single cell studies, we can study the isotropic and anisotropic secretion patterns, and hopefully in the future we can find out more mechanisms through these single cell communication profiles, cytokine profiles. Okay, yeah, so I would like to conclude here. We have done the single cell cytokine secretion profile measurement using like one type of DNA aptamer. So they are multiplexed for single cells, but are not multiplexed measurement for multiple cytokines. So what we can do next is to design the golden nanoparticle with different shape or sizes, so that we have different resonance frequency of wavelengths, so that we can differentiate different cytokine measurement in one system. That's our next step. Okay, thank you. That's all our results. I would be happy to take any questions.
So thank you very much for this nice presentation, and also here perfectly in time. So we have quite some time for discussion. So I see a few hands. I will start upstairs. Okay.
Thank you so much. Is it working? Oh yeah, it is. Thank you so much for your presentation. I had one question more towards the biological side. because you mentioned immunoprofiles based on cytokines that are produced. Did you notice any specific patterns arising? For example, cells that produce more IFN gamma produce less IL-6, something like this?
I'm not sure what do you mean the rising. So we observed when the cell interact with each other, like among between the CAR T cells and tumor cells, the cytokine are increased. If they are individual single cells, they have a lower, very low cytokine secretion.
What I meant is because you do measure in single cells, so if you can group them in different patterns, so if high producers, low producers, if you do a comparison between the single cells.
Oh, no, not yet. Yeah, that's what we want to do, but we just kind of validating our system. We have some designs using microfluidics to control even the distance between the two cells and also the pattern, different type or subtype of cells. So we haven't done that yet.
Okay, thank you.
So there was a question in the second row. It's quite some sports here for me.
Thank you, Sven. No, thank you for a very interesting talk. My question is a little bit, I mean, it follows on from the discussion we just had on positioning, actually, because I was quite curious on how you were making sure the cells were staying where they are, because you're looking at them over quite a long time. And also, how do you control the distance between the effector cell and the cell that's supposed to create the signal? Because most of these things are kind of receptor-bound situations, and so the cells need to be very close, and so it's going to be quite challenging to keep that going.
Right. This is a very good question. So right now, as I mentioned, at the current stage, we just randomly load the cells into the microwell. On the glass substrates, we have you see you have a lot of sensors there, and then we just pick up the cells. They are close to each other. Have some. So we just have a lot of cells and pick up the cells of different distance randomly. So we had a design for microfluidics control single cell positioning, but we haven't completed that step yet because we're focusing on the sensor development.
Just next one.
Yes, thank you. Thank you for the nice presentation. I had a question about the reversibility of the reaction with the aptamer. So do you use chemicals, or how do you make sure that it becomes continuous so that the gold particles can measure something again?
It's not a reversible. Actually, it's a one-time use. So it actually is a single molecule capture. So it's a two golden nanoparticle linked with one aptamer, so they capture one molecule. So whenever they capture it, we don't release it. Because we have a lot of sensors there, so the spatial temporal measurement is based on a lot of single sensors to detect it, so we don't release it after the measurement.
Okay, so you're more mapping over time, so to say.
Right.
Okay, thank you very much.
Okay, so are there more questions? Yeah, that's quite some exercise for me here. The next one I take here from the right bottom.
Thanks a lot. Very nice presentation. I was wondering about the synthesis of these dimers. So you portray them with having a single aptamer in between the two particles. And can you say something about how you synthesize that and what type of surface chemistry you need to prevent them from just sticking together?
Actually, that part done by our collaborators. So, and we kind of outsource to some company do the synthesize. We design it. We have a lab designed it. I think using this kind of, I'm not expert in that. So they using this called DNA aptamer design, the DMD, the molecular dynamic simulation. And we have more details in our papers. And then the synthesize, I think we outsourced to some company. I don't have all the details here. So we did something to measure the aggregation. Actually, sorry, I may not have all the information now. So yeah, it's not my expertise. So sorry, I just we did this kind of standardized simulation and then outsourced to some company, and we did some calibration on the aggregation on different concentrations like that.
Yeah. Okay. Well, thanks a lot. I can look it up in your papers. That's fine. Thank you.
Okay. So more questions? So I also, I think it was addressed before. So I would be interested in, so I think we worked with similar cells before, and typically they migrate over the surface quite a bit. Yeah, so they don't stay close to their target, so they just move around. So do you track them over time, or so? How is this practically done?
Because we monitor the measurement now only like 30 minutes to 1 hour or 2 hours. And as I said, sometimes we pick up the cells, they don't move too much.
Okay.
And another way is we functionalize the subjects with something to capture, for example, the CAR T cells that have to be activated by CD19. So we can also do that.
Okay, thank you. We have a question from the online audience.
No, it's actually my question.
Your question.
Online,
okay.
we don't have any questions. So
online, please use the chat.
Yeah, it's just a follow-up question on the reversibility. I think that for cytokines, monitoring increasing and decreasing concentration is very important. So I wonder what's your strategy gonna be if you want to monitor also decreasing concentration, because I think in your graphs you show only concentration increases.
Yeah. I think this is a common difficulties for real-time continuous monitoring, right? Especially when the cytokine decrease. So what we can do normally, we just using some mathematical way to measure this, some slopes, right? If we quantify the concentration over the time to the increase or decrease, we only based on the slopes of this measurement. Right now, it's no good way just have a measurement of the absolute decrease of the cytokines molecules. I think we just have to use a mathematical way to quantify it.
Okay.
Okay, more questions here. So I have another question. So when you speculate into the future, you want to, develop the assay further to some practical and medical use. So what are the steps to take towards real diagnosis towards leukemia for patients?
This is a great question. So as the other audience already asked the question, so one thing, for mechanism study to better control the positioning of single immune cells, right? So we can have a better control and then have a better mechanism studies. This can be incorporated with some microfluidic devices, which is also our expertise, but it takes some time. And the real-time monitoring as this topic thing, this is conference thing. So still, it is working in like a maximum 1 or 2 hours. I think it will be saturated still. So it's good for single cell measurement anyway, but it's not good for we also have some studies using some plasmonic sensor for tumor microenvironment on chip measurement. Then we have to just monitor the cytokine every day, but take it off every day. It's like a fake real-time measurement, but it's okay. So we just do that. So how to scale it up for like a tumor microenvironment measurement, and you can study a lot of cells communication. Right now you can see like in vitro 1, 2, 3, 5 cells maximum. Yeah. So how to scale it up for large scale measurement? Yeah, that's another thing we should do.
This is exactly what I meant. Yeah, so single cell studies are nice, but for the medical relevance, you need the global response of more and more cells. Yeah? So,
right.
Okay, so I'm afraid time's up. So I thank you again for this nice discussion as well and the presentation. Thank you. And we switch to a live presentation, actually here. So Jean-Francois Masson, I know him for quite a while. He's also one of the supervisors of one of the teams. He came over from Canada, Université de Montreal in Canada. He studied chemistry and went then for his PhD to Arizona State and did a postdoc at Georgia Tech. He develops plasmonic materials and instruments, and I also read here he founded or co-founded a SPR company quite some years ago. He received several prizes, among them also funding for Humboldt Fellowship to stay in Germany, so that's nice to hear. And yeah, he's one of the pioneers and one of the leading experts in the field of optical sensing, biosensing. He will talk about SERS, so surface enhanced Raman scattering, and biosensing with that method. So I think you need to use the mic here. Please.
Thank you very much. Of course, I can stand on the other side. So thank you very much for the invitation to be to present our research here. So I will dive into a different technique here for continuous monitoring, and something that I don't think we've seen a lot in the past. And we want to use surface enhanced Raman scattering. And the reason why is that we can create what we can call a nano endoscope. So we can create a sensor that is quite small, that could be minimally invasive, of course, and it's going to be useful for either in vivo or on-body detection. So the probes can be made actually through different ways, but the first thing is to understand a bit more about SERS to understand why it can be used for continuous monitoring. So when we have molecules binding onto metallic nanoparticles, and if you radiate that particle with a laser, you will have a very high field here, such that you can detect up to small single molecules. The vibrational spectra you collect from the SERS spectra has a lot of molecular information, and you can identify the molecule, and such that you can have spectra of single molecules, which you can also identify the molecule. These spectra are collected quite fast. It can be up to a few millisecond time scale as well. There's different ways to use SERS to be able to sense molecules. One strategy is to use targeted sensing, where you have, for example, antibodies or other molecular receptors. But that is more of an endpoint asset. That means when it binds, and we discussed that in the previous talks, you have a slowly reversible or irreversible binding on the particle. One way to do that is to use a label-free SERS, where the molecules, when it binds on the particle, it will bind transiently. So meaning it will stay there for a few milliseconds, a few seconds, or a few minutes, depending on the binding strength. And such that if the particle binds on that very hot spot that we saw on the previous slide, it will lead to a single molecule Raman spectrum. But that might unbind, and then you'll have a second molecule that will come in, and then you can sense the molecular environment dynamically that way. So one of the example I will give quite shortly is for neurotransmitters. So how can we do that? So what we use is we use pull fibers. So you can use a fiber optic or a glass rod, and you can pull it to a tiny tip. So you use like standard techniques for physiology, where you can use a CO2 laser puller, for example, and you can pull the fiber to less than a micron in diameter. Then you can coat them with nanoparticles, such that when you place them either in tissue or close to cells, if you trigger, for example, a response from the cell, the molecule can be released, and it can diffuse to the fiber tip, and then you get a Raman spectrum. So when you turn on the laser, you can monitor continuously, and here the integration time is a few tens of milliseconds. And you see, for a pure solution of dopamine, you have molecules binding on and off the particle. And the Raman spectrum that is showing up transiently here is actually for pure dopamine. And you see changes in the spectral profile because of a single molecule-like regime, which means that when the molecule binds on the surface, depending on its orientation, you'll get a different signature. It's almost like taking a picture of someone from different profiles. So that means it puts a lot of burden on the data processing, and I'll touch base on that quite soon. So now the fiber is actually quite small. So we have a tip of less than a micron. We can place that quite accurately next to, for example, neurons in this case. So we know neurons will release the neurotransmitters from very specific location on the axons. So we need to be able to accurately monitor local concentrations because the neurotransmitters will be reuptaken quite rapidly. So what we can do is we can use optical fluorescence microscopy to guide the sensor close to the axons and put the laser focus at the same place. So that's a Raman laser in this case. And then we monitor with milliseconds time scale. And then you can have, for example, a basal condition, and then you excite the tissue or the cells by pumping a different type of medium. In this case, it could be a high potassium medium, which will actually depolarize the neurons and lead to the secretion of the neurotransmitters. So we can use that to be able to monitor very, very locally in time the changes in metabolism or changes in metabolites present close to these cells or neurons. So I mentioned it puts a lot of burden on the data processing. So one thing we can do is we can build a library of neurotransmitters. So if you do classical neurochemistry, you'll oftentimes use a small electrode, and then you can do electrochemistry, and you can have very exquisite detail about the dopamine level, for example, or serotonin. But you can only measure one at a time because the potential overlap in electrochemistry allows you to only do one molecule. Since we measure the transient adsorption of the molecules on the SERS substrate, that means we can have a fingerprint of different molecules. And these fingerprints will be different, of course, depending on the molecular vibrations. So here we have a library of spectra from different neurotransmitters. So we have dopamine, glutamate, ATP, acetylcholine, and GABA. And we can measure much more than that, of course. So these are the pure spectrum. But as I mentioned before, because of the transient nature of the adsorption, the molecules will bind using different orientation, essentially, on the substrate. That means that the Raman spectrum will change in time. So we cannot use classical methods to assign the spectral signature to the identity of the molecule. So we need to use machine learning. So one thing about machine learning, it has a lot of potential for sensing, but it has also a lot of danger for sensing. So the first thing we need to do to be able to assess what kind of machine learning tools is we need to define a problem. So in our case, we need to identify which molecule is there and quantify it. There are so many tools in the literature, so you have to essentially harvest the tools and see which one works best for your application. But machine learning also needs a lot of data. It's like a beast. You need to feed it, otherwise it's going to bite you later. So you need to feed the beast with a lot of data, because otherwise it will predict poorly at the end. But since it's a black box, we need to validate, and revalidate with other techniques, with conditions we know. Because otherwise, there's always an uncertainty with machine learning, and it's not because machine learning tells you it's something that is right. So for our problem, we use the supervised algorithms. So the supervised algorithms will be quite good for classification tasks. We have a spectrum that we need to assign this proper identity. We want to know if it's dopamine present, if it's glutamate present, or if it's a different molecule. Of course, there's a number of different tasks that machine learning can do, but they're not relevant here to this presentation. So we use a convolutional neural network to be able to assess the identity of the molecules being detected. The reason is that we treat the spectrum as an image. And that image, once again, for the SERS molecule, it will be from different profile of the molecule. So we can use the convolutional neural network and train it to recognize the features that are associated with each of the molecules. Once it's trained, then we can use the inverse process where we show it the algorithm an image or a spectrum, and then we ask what is the identity of that molecule. So we use the neural networks with about 100,000 spectra to train it. So we use about 70% for training and 30% for validation or testing. And here what we have is a confusion matrix, and the confusion matrix will essentially show the proper assignment. So the diagonal line is a proper assignment for the neural network. And we get about 90% on average proper assignment for each of these neurotransmitters. So then we place the sensor next to the neurons, we measure basal conditions, and then at one point we depolarize the neurons with potassium. And what we can see is that in this case, these neurons were coming from the mesencephalon, which is a region of the brain which produces a lot of dopamine, but also glutamate. However, there's no other technique to measure them in real time and coincidentally. So what we show here is probably the first experiment where we show the co-release of dopamine and glutamate in a single experiment. So we see it because we see a number of events that show up. So essentially we treat the Raman spectrum and we ask the neural network, is there dopamine present at that time? If yes, well, we assign a value of 1 at that time. So the bars are essentially proper detection events for dopamine. And the more there's bars in the time scale, the more detection events we actually see. So what we see here is that during the depolarization, which is expected, we see a lot more dopamine and glutamate. And then we come back in the basal conditions, then we see fewer events. We can do that from different neurons of the brain, and we can see different profiles as well. But cultured neurons is not the real brain. So one step forward is to use brain slice. So what we do is we use 300 micron brain slice from mice, and then we can insert the fiber in the brain slice at a certain depth, because when you prepare the brain tissue, well, there will be a little bit of dead tissue at the surface, of course. And when you slice the tissue, you will also slice neurons. So I'm sure you will agree that these will not be livable. So then we can also modify these neurons inside in the brain slice by using optogenetic. So we can use channel rhodopsin to be able to light activate the response, the brain response essentially, with blue light, and then be able to measure the metabolite profiles at that time. So we modify the microscope to be able to do that. So we have a blue light channel now in the Raman microscope. And then we then, once again, turn on the Raman laser. In the basal conditions that we see here, we don't see very few, we only see very few events in the basal condition. And in the about 10 seconds, we'll turn on the blue light, and then we'll see a bit of a background rise, and then they'll see a pulse of neurotransmitters being detected at the same time as the blue light was shown for only a couple seconds. So the baseline should rise in about a second or two, and then we'll see just a pulse of the neurotransmission at that time. So the baseline rose, and then we'll see about now the rise of the signal that coincides the excitation with the blue light. So we can apply that for other type of metabolites. So it's not only useful for neurotransmitters. So one of the aspects that we wanted to see is, can we essentially see the metabolism of different type of cells? So what we have compared here is cancerous cells with endothelial cells, and then in that case we'll need to target different metabolites. One of the interesting aspect is that since the sensor is very small, you could place it in the tissue and be able to identify whether the tissue is cancerous or not just by the metabolism of the tissue. Or you could also see if the response of the tissue to a drug, if it actually occurs by monitoring locally the change in metabolism. So we're going to treat the cell in this case in very, very crude way. So essentially a cell, we're going to take it as a bioreactor that has glucose intake and outputs lactate, just for the sake of this experiment. So that means if we're close to the cell, we should see an uptake of glucose, and we should see an increase in lactate. So what we can do is we can use a sensor and move it around. So essentially continuously move the sensor, and what we can do is we can do an approach curve. So this is a trick from electrochemistry. So you start far from the cell, so you measure the bulk, and then as you move closer and closer to the cell, you'll start to measure more the response or the metabolism of the cell. So essentially, as I mentioned, we can treat the cell as a bioreactor with fuel, glucose, and waste product, lactate. So then we have to recalibrate the machine learning for different metabolism product. So in this case, we had pyruvate, lactate, glucose, urea, ATP, and a few more. So we retrained the neural network for that, and we had, once again, about a 90% proper assignment in this case. One thing I should mention is that we also need, in this case, to assign a class for the CO2-independent medium. So the background is always detected, and if you don't use a background as one of the classifier, it will force the background into one of the analyte, because the machine learning wants to provide an answer for each spectrum. But if you don't train the background, then it will force the background to one of the analyte, and then you'll skew the measurements. So now we can see here the approach curve close to UVEC and ELO cells. And what we can see is the gray bars are for glucose and the purple ones are for lactate. As we start far, the bars, the shaded area is for glucose. And as we move closer and closer, we see fewer and fewer glucose events. But we see an increase in lactate when we move closer. So that means we can sense the metabolism of the cell in very close proximity. For the sake of time, I think I'm going to skip the last example for protein monitoring, but we've started to monitor proteins inside mice brains, but we can expand that into the question period later on. So in conclusion, we use three types of tools. So we use Raman spectroscopy, we use nanomaterials for SERS, experimental design to be able to assess the metabolism of cells in a continuous monitoring fashion to be able to see what are the impact, for example, of drugs or for stimulation on the cells. So I need to thank the team that actually does the work, funding and Justine, and thank you very much for the invitation and for the attention.
Okay, so thank you very much, and skipping the last slide saved you for the timing. So, but maybe we can discuss over. So, are there questions? The audience, not there, up there. I bring you my
Lucky
mic.
you.
Yeah, thank you very much. Very nice presentation. I was wondering about the machine learning. Do you also need to train it on mixtures, or can you train it on single compounds and then extract mixtures?
Yeah, so we train it on single compounds because if you have mixtures, you can't really know which one you're detecting at one time point. So it's going to skew the machine learning. But then we validate with mixtures. So for example, if we train for neurotransmitters, then we can inject a mixture of dopamine and glutamate, and we should only see these two events light up.
Yeah. Oh, okay. And another question I had was about the detection of the events. You say that they are bursts of few tenths of milliseconds. What maybe I missed it. What causes this duration? Is that absorption and desorption, or is that some destruction of the molecule? Or?
So it could be a combination of both, to be honest. So we do generate a little bit of heat on the surface of the nanoparticle, and that probably facilitates desorption of the molecule as well. But it's really because the molecule moves a little bit on the particle, so it's a bit of a mixture of when the molecule binds at the perfect spot for the Raman to be triggered, and also desorption, and it could be also a bit of destruction of the molecule.
Okay. Yeah. Okay. Thanks.
Okay. More questions here? Menno?
You'll earn your pay for today.
Yes.
Thanks for your talk. Proximity is essential to generate the surface-enhanced Raman, and that means the surface needs to be accessible, and that means that fouling could kill your sensor or kill the quantification of your sensor.
Yep.
So do you see that? How do you deal with it? What's your scientific view on that?
Yeah, so this was our biggest worry when we started the project, to be honest. So we thought the sensor would work for 10 minutes, maybe 20 minutes at most, and then it would foul, and then it would be unusable. What happens, and what we believe it happens, is that we do generate a little bit of heat from the plasmonic excitation. And we've shown that we can generate a few tenths of degrees if we excite a little bit too high the plasmon. So we think the heat drives a little bit more the desorption. So that's probably one of the mechanisms. Also, we've never seen like a progressive fouling of biotiles, because biotiles could be one of the suspected compound that would biofoul the surface quite rapidly, and we've never seen any progressive background increase. So I think the biggest, the most likely hypothesis is that the slight amount of heat we generate kills the biofouling.
Even tile bonding?
Yes, even tile bonding, it seems to. Well, you don't need a lot of heat to desorb a tile. 40, 50 degrees, you start to increase the kinetics, and for some tiles, at 50 degrees, they will desorb to some extent. And what we've seen is that so one thing I didn't talk about is laser power. So we use fairly low laser power for the measurement, because at one point we tried, let's say, 1 milliwatt of laser power, and we saw the neurons actually die. So they were actually creating varicosities, which is an indication of heat generated at close to the neurons. And when we work in tissues, if we crank up the laser power, we see the tissue actually will burst. So we create essentially a micro bubble. So you need to be very careful about laser power and heat generation, and that's what we think kills biofouling.
Because heat can dissolve molecules, but heat can also induce chemical reactions.
Of course.
Yeah, and since it's such a complex mixture, it's intriguing.
Yes, it's a fine balance that we're trying to walk.
Yeah, thanks.
Okay, more questions here in the audience? I also would have, let me ask my question, then I run to you. So we talked about it, quantifying the molecules. That's a big problem, yeah? So because, you know, if you reproduce the sensor, you make the next fiber with the next coating of particles and you bring them close. Can you really, you know, quantify from cell to cell to say this cell has more, this cell has less secretion? How to calibrate such a system?
So we can calibrate because we master a little bit more the reproducibility of the sensor. So we've designed a process, and I didn't take the time to say how we do it. But a sensor-to-sensor reproducibility is actually fairly good for SERS. It's not perfect, of course. It's not HPLC type of reproducibility either. And then the way we quantify is we count the events. Because the rate of the events will be proportional to concentration. Since we're detecting like single molecule type of events, we can do digital counting essentially. And that provides us the quantification aspect. Well, I would say semi-quantification because this has a bit of a log linear relationship, which also creates a bit of higher irreproducibility. But oftentimes when you simulate the cells, the concentration change will be on the order of 10 times, 100 times. Okay. So that means that can be seen. But we wouldn't be able to see a change in concentration of maybe of 2 times. That would be more challenging.
Okay. Yeah. I come.
Thanks for the talk. So maybe it's the part that we missed out on, but I'm used to seeing SERS for small molecule and the fingerprint kind of concept. Does the fingerprint concept start to fall apart when you move to proteins and larger cytokines? Yep. And how do you sort that out?
So for us, the way we sort it out in this case here for our initial measurements is to use a SERS immunoassay. So the SERS immunoassay will use an endpoint measurement in this case. So we insert the fiber, we sense for a period of time, take the fiber out, and use a Raman tag. But in some ways, one thing that we could do is in an SPR fiber, and that could have some protein dynamic like reversibility. Well, we saw some challenges actually before in the other talks as well, and we will face the same type of challenge. But in this case here, just for just the demonstration of the concept, we use an endpoint assay.
So I would have another question. So as you know, I'm from the micro nano chair. Typically, Raman is quite bulky. So any chance to miniaturize? You showed some applications for mice. You want to have a small tool then. So how big is it at the moment, and chance to miniaturize?
So right now it's as big as you can imagine. So it's really a Raman microscope. So it's quite large. So you can miniaturize the system because the microscope part is what takes a lot of the space. If you use the fibers to deliver and collect the light, then you can create quite a small device, because then the detector can be fairly small. The laser is already quite small. So in that case, you could envision to have something that is could even be portable actually.
Okay, so we should talk about this. We also want to do a Raman in microfluidics.
Wonderful.
Okay, more questions here? Is there something online? No. Okay, so I think we are quite in time. So then I would release you. Thank you again for your presentation. We can use the leftover minute for mounting the next laptop. So thank you. So it works. So here's the microphone. Okay, and you need to use this mouse. the online audience can see it. Yeah. Your pointer.
Oh, yeah.
Okay. Good. So that's a smart one. Very good. Okay, so I'd like to introduce the final speaker of this session, Abtin Saateh. And he's from EPFL Lausanne, and you earned a 45-minute slot now. Yeah, so this is very good. So I have read he's working in also plasmonics biosensors, and he's one of the doctoral students in the ITN.
Yes.
CoSense. And I suppose you will talk about the CoSense as a whole, or maybe your part in it. Mostly your part. So that's quite nice. We will talk about long-term and continuous plasmonic monitoring. So the floor is yours.
Thank you. Thanks for your kind introduction. So good afternoon, everyone. I think you still have some energy to listen, and it's an honor today to be here. And I would like to thank all the organizers of the Sensors and the Symposium. My name is Abtin Saateh, and doing a PhD at EPFL. Well, today I will mostly focus on long-term continuous plasmonic oligonucleotide monitoring enabled by regeneration approach. So let's begin by the motivation behind this concept. So why continuous monitoring? It's actually to measure the dynamics of the chemical and biological processes. So the main benefit of this would be enabling the real-time analysis and real-time analysis in critical areas like healthcare, pharma and food industry, or environmental monitoring would basically allow us to intervene whenever needed. And the impact of this would be enhancing the disease treatment, diagnosis and the treatment, and also ensuring the quality control in pharma and food industry, or protecting the public health in environmental monitoring. So, but what is the continuous monitoring? Actually, yesterday Menno had a good already introduction for this, but well, I want to start by defining the important parameters in this sense. So the main important parameter here would be actually the frequency of the measurements. So right now, most of the biosensors are designed for intermittent measurements, and intermittent measurements are basically measurements with kind of sparse measurements, and they just give you some number of the biomarker, I mean, at that measurement time point. However, if we increase the frequency of the measurements, we could capture actually not only capture the value of that biomarker, but also capture the trend of the fluctuations of the biomarker. So to achieve higher frequencies, we need to do measurements with higher frequencies, and in the frequency discussion, first we need to analyze the response time and the reversibility time that would in total give us the total measurement time of the this part. So for any biosensor, any readout method, we have a measurement time that it could be first stabilization time, then measurement time, then the response time of the sensor. And then for the continuous part, we need to have a resetting time or reversibility time. So when coming to discussing this reversibility time, we need to make sure that the sensor comes back to the baseline. Because as a sensor comes, if the sensor doesn't come to the baseline, it cannot provide accurate and precise measurements. And in that sense, repeatability is quite important, so that for same concentrations of target, we need to see the same precision over and when it's injected or measured over and over again. And when actually discussing this over and over again, we need to check the long-term performance. So apart from usual criteria in any biosensor, we need to, for any continuous biosensor, we need to also check these parameters. So there are different methods to do this, but well, we chose to go with plasmonic biosensing. And plasmonic biosensing, specifically with surface plasma resonance, which is considered as the gold standard of biomolecular interactions. And when the analyte comes close to the surface, we can see a resonance shift in the tip of the spectrum. And using gold nanoparticles, we can actually enhance this sensitivity even further and do more sensitive measurements. So in our bioassay, we first started with, in our work, we developed a gold nanoparticle enhanced SPR assay. So firstly, we achieved a picomolar range of detection for 20 by using 20 nanometer gold nanoparticles. And while doing this, we paid special attention to decrease the blank signal. And as you can see, the blank signal was quite minimal. And then we established the calibration curve and the whole range, and we also checked for the SPR. SPR is actually quite known, SNR, it's similar to SNR, which is known in signal processing. And here it's defined as the specific signal to blank signal ratio, and the blank signal is basically the measurements where we have everything but just the target missing. To compare all different gold nanoparticle sizes in the assay, we checked from 20 to 40 nanometer, and we checked their calibration curves. Although, well, we reached better sensitivities with 40 nanometer gold nanoparticles, and we also checked and compared it with a label-free bioassay where the limits of detection and the working range of the sensor is almost 3 orders of magnitude higher. But comparing all these different gold nanoparticles, we can see that 20 nanometer gold nanoparticle actually achieves a better dynamic range and SPR. That's why we chose to go on with the 20 nanometer gold nanoparticles. And coming to the reversibility, which is the most important part of this work, we used regeneration assay. And in this assay, well, the step 0 and step 2, both of them are just having the buffer, and the main parts is the step 1 and step 3. Step 1 is where we inject our target sample, and step 3 is the reversibility time, and where we introduce regeneration buffer. However, in any regeneration-based approach, the most important part is to make sure that the capture molecules on the surface are not being deteriorated, and we don't have a loss of performance. So first, initially, we checked the three same concentration of the target, which is 1,000 picomolar. And here you can see the response time of the sensor is consistently 4 minutes and 1 minute reversibility. And well, for all of them, it's the same response, and reversibility has been repeated. We also checked under SCM for the gold nanoparticles and how they bind. But, well, that's just the beginning. To make sure that the regeneration is optimized, we use DOE method to check different buffer types and their concentration, the different salt types and their concentrations, to make an optimized regeneration buffer that would help us both for optimizing the label-free and gold nanoparticle enhanced regeneration buffers. Another important criteria for a continuous biosensor is, of course, the temporal resolution of the sensor. Well, by tuning the flow rates, we could, of course, change the detection time and the response time of the sensor, while the reversibility time or regeneration time is fixed as 1 minute. However, the trade-off between the flow rate and the signal intensity is that as we decrease the detection time, the sensitivity would be reduced. However, this flexibility in tuning these would be basically give the clinicians the flexibility to tune how sparse they want to monitor the biomarker of the interest. So, coming to the regeneration buffer, which was optimized, we checked for long-term continuous monitoring capability in buffer. And here we had more than 100 times of measurement with 5 identical concentrations of the target. And the measurement is a day-long measurement. Well, for example, in 3 of these 5 cycles, you can see that in the bottom left panel, the measurements, the responses and the reversibilities are all quite consistent. And in the right bottom, you can see, maybe I would check the laser. In the right panel, you can see the overlay plots of all these concentrations, where we achieved CV of around 3%. Another parameter is the long-term stability of the bioassay. We checked the stability of our bioassay in using regeneration, where we just injected the identical concentrations of the target over 9 days. And here you can see the CV was less than 4%. Another important criteria for any biosensor is, of course, the capability of working with under complex matrices. In this case, we used whole human serum without any dilution or filtrations. And we had a detection in picomolar range, which was quite similar to our buffer case. And we also checked the long-term continuous monitoring in whole human serum. And this is the result of 1 day long experiment. And the overlay of all of them, which we achieved coefficient of variation for these concentrations of less than 5%. So in summary, we established a regeneration-based reversibility approach, which was working in both buffer and serum. Then we checked for the binding performance to ensure the assay has quite minimal blank levels and with the maximum binding capability. And also long-term continuous monitoring with minimal coefficient of variation and 1 day for the whole monitoring and the bioassay stability of around 4%. And efficacy in complex matrices. So before concluding, I would like to take a moment to acknowledge the contributions of the BIOS lab and the support, their support during this research. Specifically, I want to thank my supervisor, Hatice, and all the colleagues in BIOS lab, specifically Said and Jaray, who helped in this during the experimental parts, and the MBX lab for their opportunity of the secondment, specifically Peter and Livio, and the Consensus Consortium for the funding and, of course, all the meetings during this time, which gave us quite valuable insights. And well, finally, I would like to extend my gratitude for all the people present in this meeting and this symposium. So I look forward to your questions. Thanks for your attention.
Okay, thank you for your presentation.
Thanks.
And actually, I'm surprised, yeah, because somehow we earned some time now, because we have time till 6 o'clock. So half an hour time for questions for you. No, just kidding. So
Yeah.
do we have questions here from the audience?
Thank you very much for the presentation. Very interesting. So I want to go back to the flow rate experiment. So you said that there was less sensitivity at higher flow rates.
Yeah.
Is that solely due to the shorter time, or is there something in the fluid dynamics that makes it less sensitive?
Well, mostly the shorter time for the detection, and actually the since I was using the same sample volume, so there wasn't enough time for the binding, and that's why we had less signal, binding signal.
Okay, so then presumably you could use even the low flow rate, so less sample volume, and still achieve shorter times.
Yeah.
Okay. And so when you regenerate, sometimes, the stability was quite impressive, but you see some long-term fluctuations, which seem to appear. Is that an artifact, or is that something that you would expect?
We saw it in buffer experiments, a bit of basically the response, which was sometimes going up and down, but well, we are not really sure about the reason, but basically when we put all of them over as an overlay on top of each other, the CVs were not really a big problem. Was around 3% in buffer.
Okay. Thank you very much.
Thanks.
Okay, so more questions from the audience here? Menno, I expected that.
Hi, Abtin. So this is an oligonucleotide assay, right? And you tested it in human serum?
Yes.
Did you show that?
Yes.
A logical next step would be, like what Professor Aili has also shown, is to detect proteins and develop regeneration methods for proteins. Did you go into that, or is it really a next step for someone else to investigate that?
I actually investigated started to investigate a couple of months ago with thrombin case, but there I was also using the aptamers for that. So to make the assay and everything quite similar with the current assay, but extending it the same approach toward protein, long-term protein biosensing.
Okay, yeah. And what are you finding in terms of type of buffer that you would need to do regeneration? Do you get into the same parameter range, or?
Actually, for the aptamers, it wasn't the same regeneration buffer, and I had to optimize this the buffer. But actually, I haven't checked for another different types of aptamers yet.
Mm-hmm.
But for the thrombin aptamers, it was a different one. Just with current one, it was not really working well.
Okay, thanks.
Okay, do we see more questions here in the audience? Everybody got tired. Online questions? So then from my side, the same question again. So how large is the setup at the moment, and is there a chance to miniaturize that?
Well, the setup is around 45 by 45, around something like this. Mm-hmm. So it's not a wearable device. However, with the current setup, what we envision is basically, since in a case of critical cases, like for example for a patient who has inflammation, they are already in the bed settings and clinical settings, and there is not for such a condition, there is no need to miniaturize, and with the current setup, it could be used. However, there are already miniaturized SPR devices, like the company which Jean-Francois has. There are also some other companies, and you can find quite miniaturized SPR setups. But using regeneration, you cannot really go regeneration with buffer. You cannot go for a variable, but for settings like environmental monitoring or industry in the food or pharma industry, or patient in the bedside in the clinics, it could be used.
Okay, thank you. If I don't see another question, I would ask another one.
Yeah.
So we still have time. So we should not release you less than 10 minutes, I would say. So CoSense, this is an ITN.
Yes.
Yeah. So
Yes.
and are you first generation or second generation?
I think we are the first.
You are the first generation. Yeah. So. 15 PhD student, and then the next phase will come, right? No? You don't know yet. Eventually, yeah. So this would be my question into the future. So I assume I have seen quite some results. The PhD is near.
Yeah.
So what would be the next step in this project? So how could you expand that? Okay, we heard about proteins, yeah,
yes.
But I don't know. Is it the idea to also to go multiplexing, or what is the application you see?
Of course, in this case, the first step would be to extend to the protein biomarkers. And, for the multiplexing, there is a need for, especially in the case of, for example, inflammation that we just talked about. However, after proving efficacy with proteins, it could be definitely extended toward the multiplex biosensing, and not in a continuous mode, but in rather endpoint. I'm already also doing the multiplex biosensing, which we just presented in consensus meeting. Yeah.
Okay, thank you very much. So I don't see more hands. Then we earned a longer pause, I think. So there are plenty of posters outside to check. Yeah. Poster session is coming, food is coming. So there's also another session coming with all the sensors teams coming in. So the room will be more crowded later on. So for the online audience, nice that you were in, but we haven't seen or heard you. So there was no question at all. There's one coming. Wow, okay. You saved the online crowd. Yeah, so please.
Yeah.
So Hossein mentioned it was a great talk, and they are wondering if what is the maximum time you are able to perform real-time measurement in serum, and how do you deal with the biofouling issue?
For the measurement, regarding the measurement time, the longest that we did was actually 1 day long experiment. And for the biofouling, we investigated quite a bit in the first parts of the assay, which today I didn't have all the results regarding that much. But before going to serum, we investigated with different PEG lengths and their concentrations. What I used was basically a thiolated PEG.
Okay, no more, but now you also mastered the 10 minutes question and answer. So thank you very much again for your presentation.
Thank you.
And I also like to thank all 4 speakers for this first session, and we reconvene also for the online audience at 19:45 CEST, which is quite a long pause now. So thank you.
Thanks.
All right, everyone, welcome back to the final session for our continuous sensing workshop and conference. So I'm Mike Danielle from NC State, and I have the pleasure to present the 3 final talks and then sort of closing session as my team walks in appropriately late, so wave to them. And I have the pleasure to first introduce Professor Tom So from Stanford University. He'll be presenting over a video. Unfortunately, he couldn't make it, but I could tell one brief story, which is he continually poaches my undergrads and master students. I now have 3 that have transferred over to his lab. So I'm hoping to collect some type of fee in the future for hopefully delivering good students. But many of you might know Professor So is professor of bioengineering and electrical engineering at Stanford University, and he develops different types of bioreceptors and biomolecular recognition systems. And tonight he's going to be presenting his talk on continuous in vivo biomarker detection. So without further ado, I think we can play the video.
Hi, everyone. I hope you're all having a great meeting. I apologize for not being able to be there in person. First and foremost, I would like to thank Professor Menno Prins for putting this wonderful venue together on this very special topic. My name is Tom So, and I'm a faculty member at Stanford. And today I'll tell you our work on creating continuous biosensors for measuring molecules. And Menno has asked me to focus really on the optical part of this technology, which I'll do. I think for this crowd, I don't have to spend too much time, but just to motivate our work for important machinery that our society has created, like a jet airliner, we take great care in monitoring its state. So for an engine, a jet engine, we measure temperature, pressure, vibration, etc., continuously. And that information is fed back to the engine manufacturer, who have monitoring stations to measure its state to make sure disasters don't happen. But for the most important machinery, that's ourselves, we do quite the opposite. We check once a year if you're diligent, or we only check when you know there's something wrong with the machinery. And so many years ago, we thought, in the future, wouldn't it be amazing if we could continuously measure our molecular state in vivo, in real time? And so the reason why we can't do this now, of course, is the reason for our conference is because we don't have sensors. And if you take a step back and think why we don't have sensors, is because continuous in vivo molecular measurement is really hard to achieve because we can't do any sample preparation. So for example, when we do in vitro diagnostics, we add reagents, we perform washes, we exchange buffers, etc. But we can't do any of that in vivo, obviously. And the sensor has to work continuously in a challenging environment, such as blood and tissue. And this is, in essence, the reason why continuous molecular measurements are really difficult. And again, I think to this crowd, I don't think I need to dwell on this. There are only a handful of molecules that you can measure continuously. One's a pulse oximeter, which has no chemistry. This is a simple laser spectroscopic analysis. And the other, the famous one, is a continuous glucose meter, which took more than 40 years to develop. And this is possible because there is a naturally occurring enzyme called glucose oxidase that essentially converts glucose abundance into a measurable electrochemical signal. And without that enzyme, this wouldn't have been possible. But the problem is that these strategies are not general. You can't measure hormones or metabolites or drugs using these approaches because they don't have sufficient optical signatures, and we don't have enzymes that'll convert them into a measurable physical signal. So we thought many, many years ago that it requires a very different way of looking at this problem. So today I'm going to tell you really three different things. First, we'll start with aptamer-based biosensors. Then I'll talk a lot about how to create these aptamer switches. And then I will talk quite a bit about sort of our recent work in creating molecular switch-based biosensors that are not based on aptamers. So let's start here. So our initial work, more than 10 years ago now, started with an electrochemical measurement, and it was really spearheaded by a very talented grad student, Scott Ferguson. And the goal of this project was to come up with a quote-unquote general approach that can measure many types of biomolecules in vivo in real time beyond glucose. And the central idea was to use these molecular switches that bind, usually undergo a conformation switch, and give you a signal. So this is Scott's hand holding the first chip, and it took the form of a microfluidic device. And inside the device, we integrated three different technologies, which I'll briefly go over today, to achieve continuous measurement. And with this device, we're able to dope in a drug. This is a chemotherapy drug called doxorubicin into human whole blood for many hours. This is typically the sort of duration of an infusion. So doxorubicin is an infusion drug. And human clinical concentration is about 1 micromolar, which we're able to detect. And so we're able to go up and down without any sample preparation. And this is because the aptamer switch is reversible. So if the concentration drops and there's less dox, it'll revert back to its original conformation. So that's in vitro into spike samples. This is the first continuous drug measurement out of a live animal model. So this is pharmacokinetics in real time, if you wish. So we give a bolus injection, you can kind of see how the drug is distributing itself. And this strategy is quote unquote general because you can flip and swap out the aptamer switch to measure other things. So for example, this is another small molecule drug, and we can measure that as well, where we can keep the hardware pretty much the same and just swap out the switch. And in that work, we learned that folding really is the key. And so for this sensor, we sort of tinkered around with the aptamer configuration so that at clinical concentration, we get sufficient gain. So we're in the steep part of the curve as we wiggle around the clinical concentration. And Scott performed this key experiment, which is to challenge this switch with other molecules. So these are small molecule drugs that bind to DNA. And as you can see, at much higher concentration, they don't give you any signal because non-specifically bound molecules don't cause the switch to fold. And this is where we realize that folding is really the central idea here. So we took that idea, over the years much further. Now, what we have done more recently, is to use nanoporous electrodes that we can create through. So these are nanoporous gold electrodes that we create by co-depositing silver and gold and annealing them in different conditions and sacrificially etch away the silver to create these nanopores where we can hide our aptamer switches. And the idea here was to protect them from non-specific binding and enzymes that could clip these aptamers. And what we wanted to do is to create a probe that we can insert into specific tissues, in this case, tumor tissues, and measure drug distribution within the tumor tissue itself. If you think about it, it's kind of funny, when we dose drugs, we measure drug levels in blood. And we do that because that's what we know how to do. But unless it's for example, a blood-borne cancer like leukemia, that's not really relevant. What's relevant is the drug concentration within a tumor. And we didn't have the technology to measure that directly. So this is what we created. So this is an insertable probe. You can see there's 3 little dots here. Those are 100 microns by 100 micron electrodes. And if you blow them up, they have a morphology like this. These are the nanopores that I just described. And of course, inside them, there are trillions of our aptamer switches all over the place, of course, that you can't see. And then what we do is insert this flexible probe. It's a polyimide-based polymer probe inside the tissue of a rat model. And as you can see, this is the center of the tumor. This is the surface of the tumor. These are 3 different locations. So obviously, when you inject the drug, it'll hit the surface of the tumor first. That's channel 3 that you can see. It hits in time first, and you get the greatest concentration. And the center would arrive much later at a lower concentration that you can see. And by stitching this together, we're able to get pharmacokinetics in the tumor tissue and measure the drug concentration in real time. And so this is the first time we're able to do this. And what's really satisfying is that, as you can see, the pharmacokinetics in blood is vastly different than what's actually happening in the tumor. So this capability will now enable us to study sort of drug distribution in tissues. And we are now planning for human clinical trials using this type of technology. And, we started with electrochemistry, which I think other presenters here, including Kevin, have talked about. And it has many advantages. And I think one of the key advantages is integration with circuitry. But we also started to investigate photonic detection because it offers key advantages. Our lab actually works on both electrochemical and photonics. And what photonics offers is that you don't need electrodes, so that's different. You have multiple colors. Unlike current, you have different wavelengths to play with. Typically, you have more sensitive detectors. For example, detecting single photons is not that difficult. You can buy a single photon detector for a few $1,000s, whereas single electron detectors would be much, much harder. Most importantly, you have these proximity effects like forced resonant energy transfer, where you get a signal only when dyes get close to each other, which we take a lot of advantage of, which I'll describe today. So we started to investigate in this area. So I'm going to show you a few key recent work. So this is work headed by Amani Hariri and Alyssa Cartwright and Konstantin Dori. And what we try to do is to play a similar sort of game for continuous optical detection of small molecules in complex media. And so this is, so for this type of work, we come up with an aptamer switch design. This particular design is called a bubble switch because there's a bubble in the middle. And basically, it's a internal competition-based architecture where you have the aptamer, which is the black part here, an internal competitor, and you could tune the strength of this internal competitor by tuning the length and sequence, meaning you could put in mismatches, and you put in fluorophore and quencher. So typically, when the switch is not seeing the target, it will be in equilibrium in these two states. But when there is the target, then the equilibrium shifts to the right into this target-bound state, where you get a signal because the fluorophore and quencher will now be further apart from each other. As I mentioned, by tuning the length and sequence of the competitor, we can change the sensitivity of the switch by about an order of magnitude. And what's also really interesting, and this phenomenon is not fully understood, is that by putting in mismatches, we can actually change the temporal resolution of the switch, which is fascinating. As you can see, this switch is much faster than, for example, this switch. So this gives us the capability to tune the sensitivity as well as the switching time. Then what we did is to develop a really useful optical detection hardware. So it's a busy slide, but the key here is to use evanescent field. So as you know, as you guide light through a waveguide, like an optical fiber here. So here we're using a optical fiber that's been pulled to create this evanescent field near the tip. And as the light is guided, there'll be a little bit of light that leaks out. It's called the evanescent sort of field. It's about on the order of 100 nanometers. And because that field is limited in sort of volume or depth, it doesn't pick up any signal outside of that. So it's a nice filter. So autofluorescence from blood and other things, it'll reject it. It's a filter. And so we launch the light to excite the fluorophores, and we also detect the emitted light onto the waveguide that comes back. And we used very sensitive sensors. You can see that there are 2 single photon counting modules along with the lasers and filters. That way we are able to do very sensitive detection directly in complex mixtures without having to worry about background fluorescence from the media. So we're able to achieve, so this is for measuring dopamine, pretty stable and sensitive detection of dopamine. This is in buffer. Here's 1 micromolar signal. And for about a day, it's pretty stable. And we, of course, care about complex mixtures. This is cerebrospinal fluid. And again, it's stable. We can measure down to 500 nanomolar over a relevant period of time. So that's where we kind of started with optical sensors. Then we realized that there's no need to do one-dimensional measurement like a fiber. Two folks in the lab, Chan-Ho Park and Ian Thompson, partnered up to come up with this very clever idea of doing continuous 2-dimensional measurements. So the title of the talk is Real-Time Spatio-Temporal Measurements of Extracellular Signaling Molecules using an aptamer switch conjugated hydrogel matrix. So the motivation was really this. You know, the invention of the green fluorescent protein many, many years ago by Roger Chen and others enabled us to measure proteins that are expressed within the cell. But so it's used extremely, widely, but there are not many technologies that allow you to measure molecules that are secreted outside the cell in real time continuously. So this is what this technology allows you to do. And Chan-Ho and Ian named this technique semaphore, which I thought was a good name. And the idea is to embed our molecular switches inside the hydrogel matrix. So they're everywhere. And what we're trying to do is to detect secreted signaling molecules. So as a model, we're using a very well understood system called Dicty cells. These are amoeba. When they are starving, they secrete cyclic AMP to tell each other to gather, and they do really incredible things to go to a different region where there's more food. But anyway, so the idea was to detect secreted cyclic AMP with our aptamer switches and to distribute these switches everywhere. And using that, we're able to observe individual cells in space using bright field and also in chemical space in terms of concentration of cyclic AMP. So this is bright field. So as you starve them, they will talk to each other, and then they will start to gather, as you can see here. So this is bright field. This is in chemical space. So what we did here is to inject cyclic AMP, and now you can image this same field of view in cyclic AMP concentration space. And so, of course, they will start to gather where the concentration is highest, as you can see. And then what we do is we track individual cells to see how they're moving. And when you do that, it's quite incredible. So this is sort of the graph of how they're moving. So indeed, sort of the vector direction is centered around where the concentration is the highest. But you can kind of see biology is never digital. There's quite a bit of sort of distribution, and I think they're doing this to create diversity for themselves. Anyway, I found this very interesting now that we could sort of track individual cells and see the spatial as well as chemical information. And what's even perhaps more interesting is that we can observe coordinated cell movements in space and chemical space. And so what we did here is now this is sort of larger field of view. We can study many different regions. There are 3 different regions to study. And it was very well known that these amoeba, they coordinate in time. So this is a very well-known 6-minute sort of wave period. And we can sort of see in different regions, that indeed that's what happened. But anyway, this ability to now measure continuously specific target molecules in 2 dimensions in real time, I think can open up discovery of new biological phenomena. Okay, so obviously this aptamer-based switch idea has many potential applications, both in basic science and potentially clinical. But at the end of the day, the core comes down to aptamer switches. Whether it's electrochemical, optical, it doesn't really matter. The capability of the sensor will come down to how good the aptamers are. So I'll tell you sort of our work on how to make these aptamer switches. So let's switch gears and talk about aptamer generation. So as you know, evolution is powerful. We just need 3 things: mutation, selection, and preferential amplification of the winners. You can do this in a lab. And these are the 3 folks who really opened up this field of chemistry. And I think the big credit goes to George Smith, who invented phage display many years ago. We use these principles, but we actually do selection on nucleic acids. And so I won't dwell too much time here. When we chose nucleic acids rather than proteins, because they're much more manageable to evolve molecules with functions beyond just binding. We're interested in switching, of course. So over the last, say, almost 4 decades, when this work started, people have sort of followed this workflow where you create a large library, you do affinity-based selection, you analyze the sequences that survive the selection, and you make a few molecules that look promising, and then you characterize them. So this is sort of the process of SELEX that has been sort of in practice for almost 40 years, where you do bead or filter-based selection. You clone and sequence through E. coli cloning, and you look for consensus sequences, thinking that those are the aptamers that have the features that you want. You make a few and you test them. And when you carry out this process, you realize that a lot of this you carry out sort of blindly. So we didn't like that. So I guess about 15 years ago, we used high throughput sequencing to analyze the pool that was enriched and predict winners and biases that way. Of course, this is now the common, most widely used way of doing selection. I'm going to tell you a little bit about today how we created the technology to whittle down a large number of aptamers into a few. And most recently, we invested a lot of time and effort in how to do affinity and specificity measurement in a massively parallel manner using sort of the Illumina sequencing technology in new ways. So, I want to pay attention to time here. So when you do selections from large libraries, you're really limited by the sort of fundamental limitations of mass action. And the enrichment that you want to do for the, let's say, high affinity aptamer, if that's what you want, is limited by the ratio of KDs. So, this is why typically selects necessitated many, many rounds of selection. But in every selection, you have a lot of biases. For example, PCR bias that comes in. And at the end of the process, so typically you would do like, say, 10 rounds of selection, and each round you do 15 rounds of PCR. You have 150 rounds of PCR built in. And so the aptamers that come out do so not only because they have favorable binding properties, which is what you want, but they also have these biases, and you can't separate the two. So we started to think about a different way of generating aptamers that can measure the affinity and specificity of a large number of aptamer molecules and sort them individually. And we now call that particle display. And this is really, really spearheaded by a very talented electrical engineering student who never pipetted before joining the lab. So this work started about 10 years ago. And the idea was to convert solution phase aptamers into aptamer particles. So aptamer particles are 1 micron polymer particles that are displaying 100,000 copies of one sequence, so they're monoclonal. And we do that through emulsion PCR. I'll sort of skip the details here. And then you label with target. And of course, the aptamer particles that have high affinity will be brighter than those that have low or no affinity, and we sort that out in FACS. So this now has become a screen from a selection. So it's like going from phage display to yeast display, so to speak. And by doing that, we can now kind of see individual aptamer particles, and using FACS, put a sort gate around it and sort them individually. And by doing that, we're able to generate aptamers fairly consistently for easy targets like thrombin, and even tough targets that was really hard to make aptamers for. But the real power of the technology comes in specificity, because we can label the target with one fluorophore and the background or non-target, so to speak, with a different color fluorophore. And because FACS can sort particles based on multiple colors, you can now sort of profile the affinity and specificity of every aptamer particle. So what I mean by that is, let's say we label the target with a green fluorophore. So you want to put a sort gate around high green on the x-axis here. And we can label, shotgun label serum proteins, for example, with a different color fluorophore, say red. So you want high green, low red, and pick them sort of individually. So it's not counter selection, it's fundamentally different. This is individual profiling of affinity and specificity. And by doing that, we're able to create some incredibly good aptamers that operate in complex media. And sometimes, not always, we can generate aptamers that can outperform very good monoclonal antibodies. Anyway, then we began to think, why are we trying to be sort of just as good as antibodies? Can we do things that antibodies cannot do? But the problem with RNA and DNA is that, their chemical repertoire is somewhat limited. And the idea was, can we endow them with additional chemical functionalities that don't occur naturally? And this idea of non-natural aptamers have been around for a long time, almost as long as aptamers themselves. But the way people approached it is by synthesizing non-natural nucleotides and then engineering polymerases to incorporate them into the aptamer. But this is really hard because polymerase engineering is like telling the polymerase to take these non-natural things, copy them, and make no mistakes. And so, of course, on top of that, you can't make big changes because polymerases will choke as it's trying to read through it. So we thought about how could we add any functional group, including things that are just not even biological, without having to engineer polymerases. And the idea came about with Jia Nu, was a postdoc at the time, by incorporating non-natural nucleotides with a click handle. So we can, for example, swap out all the T's with a non-natural U, and then you put a ZID handle to any chemical functional group that you want. You click them on, and it turns out that reverse transcription is much more sort of loosey-goosey. It doesn't really matter. And so you retain the ability to do amplification with anything that's clicked on. And so just to demonstrate this, we start to make man-made, made-up molecules. We call them sugar aptamers. So we wanted to target a lectin. So lectin is a sugar-binding protein, this particular one, and it's a mannose-binding lectin. So we started to now click on mannose onto our DNA backbone and evolve using our multi-parameter particle display so that preferentially this pool is evolving to bind to just one target, our target lectin, and none of the other lectins. This is a lectin array. So we now have this ability to evolve, quote unquote, or amplify these non-natural aptamers, and also have this capability to do, specificity and affinity profiling individually. We've pushed this a little further. So targeting sugar receptors is one thing, but targeting sugars themselves is a much harder and much more sort of important problem. So I won't dwell too much time here, but using pretty much the same trick, but now adding a chemical functional group that have natural affinity towards sugars, we can now target just the sugar part, the glycan part of a protein, and not bind to the protein using our technique. And so here it's you have a target, same protein that's glycosylated, so it binds with good affinity. If it's not glycosylated, it has much lower affinity. And so we're really trying to use aptamers to do what antibodies have trouble doing. So I think that's really the future of aptamers. Anyway, what is really exciting for aptamer discovery technique is this ability to now display many, on the order of million to 10 million aptamers, both natural and non-natural, on a flow cell surface. So this is work really spearheaded by these three talented folks here, Diana, Trevor, and Peter. I'll skip the details so we can talk about other types of, oops, this keeps coming up, optical switches. But now using this type of massively parallel aptamer discovery technique, again, we can create aptamers for functions that antibodies would have a hard time doing. So here what we're trying to create are aptamers that would recognize small molecules that are chemically extremely similar. So for example, between these two, there's only a single hydroxyl difference, same here, etc. It would be very difficult to make antibodies that could have that level of specificity. But with this type of aptamer screening technology, we can now create aptamers that only bind to this and none of the others. so on and so forth. And finally, we have started to put together a design principle about making them into switches once you have found these aptamers. And we are learning now there are many topologies of aptamer switches, and this is one of them. These are switches called ISD switches, or intramolecular strand displacement. This is work headed up by Brandon and Amani, who you saw before. And the idea here is to, again, build in internal competitors, and by changing the length and the sequence, you can fine-tune the strength of the internal competitor. And also by changing the linker length, you can control the quote-unquote effective concentration of the internal competitor. So these are now two knobs that are not orthogonal, but they're not perfectly aligned either. And by tuning them, we could certainly tune the sensitivity starting with the same parental aptamer. But also, when they have the same sort of thermodynamic or sensitivity, we can make switches with different kinetics, which is very exciting. Anyway, so that it kind of wraps up sort of our aptamer portion of the talk. But so there's lots of really cool and useful things you can do with aptamers and aptamer switches. But even for us who work in this area, it takes a lot of effort, expertise, and resources to create these probes. So we started to think, can we make different types of molecular switches? Because, you know, we're interested in aptamers in this context, not because they're just aptamers, we're interested in switching. So we started to think about, could we use different types of molecular switches altogether that's different from aptamers? So I want to talk about our recent work on antibody and protein-based switches. So I'll start with this work headed by Ian. So it's the same Ian that you saw before, and Jason Saunders. It's an antibody-based molecular switch for continuous small molecule monitoring. So the idea was this. Antibodies are awesome. They're available for many targets. They're highly conserved structures. So this is an IgG. Of course, the problem is that they don't switch. And what Ian and Jason came up with was, hey, what if we, again, make an internal competitor? So the idea was to have a DNA linker to the bait that will bind to the Fab region of IgG, but label the antibody and the linker with dyes that do forced resonant energy transfer, FRET. As you probably all know, FRET is a very proximity-sensitive effect. So when there's no target, typically the switch will go between these two states, mostly on this side. And when it's closed, you'll get a high FRET ratio because they're close to each other. But when there's the target, the equilibrium will now shift to the right. And because this is occupied, the FRET ratio will drop because they'll be further apart. So that was the basic idea. And it took a while to synthesize these constructs, but Ian and Jason made a switch for cortisol, a very, very important master hormone that regulates our sort of not only stress response, but our sort of, it's diurnal, so it has many, many uses in the body. And so they made the switch, but the problem was that it wasn't very sensitive. So EC50 is about 110 micromolar. In blood, this is sort of the physiologic concentration. Unfortunately, it's in this flat part of the curve. So this is normalized FRET ratio. And so this wouldn't be a very useful sensor. So that was a little disappointing. Then, of course, Ian and Jason came up with a really good idea on how to shift the curve to the left. And that is to use mismatched bait, or a bait that is not cortisol, but there's something like cortisol that have lower affinity to the Fab region. And as you probably know, cortisol has many sort of metabolites, and one of them is corticosterone. So, for example, rodents, they don't produce cortisol, they produce corticosterone. It's chemically very similar. And this antibody has affinity to both cortisol and corticosterone, but the affinity to corticosterone is much lower, so you have a weaker bait. And by doing that, we're able to switch at a much lower concentration. As you can see here, you've almost 2 logs. That's smack in sort of the sweet spot of physiologic concentration for humans. And this switch works very well. In buffer, we take a little bit of a hit, but it works directly in 100% plasma as well. And so then we used our sort of near field based detectors that I described earlier, where we launch light, excite the dyes. We also pick up the emission from FRET, goes into our single photon counters. And by doing that, we're able to now continuously measure cortisol. Down to, we get pretty good signal to noise, even down to sort of 10 nanomolar. Just for calibration, this is it's more than 1 million times lower than what a CGM could do. And these are sort of individual on and off kinetics of our switch. And it's fairly stable. So I think, this type of switch has a promising future for applications. I'm I'll show one more switch. That's right. And so finally, we start to think, could we take advantage of both aptamers and antibodies? And the idea is this. So typically in the both aptamer case or antibody case, we're asking one molecule to do a lot, which is to bind specifically with high affinity and switch and give a signal. So we said, hey, why don't we actually create a chimera of antibody and aptamer where we give them a break. We tell the antibody to do its thing, bind specifically to a target. And then we tell the aptamer to do what it's good at, which is you don't have to have good affinity or specificity because that'll be done by the antibody, but when it's bound, switch and give a signal. So we're trying to distribute the workload, so to speak. And by doing that, it works in complex mixtures and it gives you much higher sort of sensitivity, clean signal down to 1 nanomolar. And so I think, but this is all enabled by these sort of proximity effects of the dyes, which are quite a powerful feature of optical sensors. So with that, I want to be cognizant of the time. I want to sort of wrap things up. So where do we want to go with these capabilities? We live in this world, but I imagine in the future, we will be able to, especially this community, will be able to create technologies that can measure many things continuously in vivo. And by doing that, that kind of opens up the door to measure problems before they occur. For example, if we could measure cytokines in real time, we might be able to pick up infections before any symptoms arise. If we could measure insulin continuously, you could pretty much solve type 1 diabetes. And so there's lots of examples where we could really change the model for healthcare by keeping them healthy and not try to fix them when disaster has happened. And if we can do that, we can sort of flip the model, so to speak. I think this is a better way of tackling healthcare problems. So I want to thank our very, very talented group of young scientists and engineers from many different backgrounds, coming from synthetic organic chemistry all the way to very advanced hardware design. I want to thank our funding agencies, a special call out to the Helmsley Trust for being a staunch supporter of this vision. And I'm afraid I won't be able to take any questions, but I wish you all have a fantastic meeting. Thank you.
So unfortunately, there won't be any questions, but we can move on to our second speaker and our third speaker who are both here. So we will have some extra time for questions maybe for them. So while she gets set up, our next speaker is from Dynamic Biosensors. Anahi Higuera Rodriguez is coming to us to talk about the kinetic FRET assays to study binding-induced conformational changes of nucleic acids and protein-protein interactions by ternary complex formation on a switch-sense biosensor. That's the title. Now I take a quick breather and let you guys know she comes to us with kind of an interesting background, doing her degrees at Leuven and University of Grenoble in nanotechnology and working on developing different types of sensors, but has a very strong interest in entrepreneurship and has developed sensors at a couple different companies. And now she's with us today from Dynamic Biosensors. So without ado, please.
Hello. Okay. Thank you. Yeah, my title is a little bit long. Sorry for that. I'm really happy to be here, so thank you for the opportunity. And basically today I'm going to talk about some FRET-based assay to look at conformational changes of nucleic acids and also to look at ternary complex formation. And this topic about kinetics has been touched in the previous presentation, but I just want to give like the basics. So when we have two binding partners, when they come together by non-covalent interactions, they come together with an association rate constant or K-on, but these can also fall apart at that dissociation rate constant. And when we have the ratio of these, we have the equilibrium dissociation constant or affinity value. So when we have two different interactions with the same affinity, let's say 100 nanomolar, they can all actually be the result of very, very different kinetic pathways. As you can see, they can be the result of very fast kinetics, so very fast on and off, or very slow kinetics. And these we cannot actually observe in a normal titration assay, so we would actually look at them and they would be the same. So that is why it's important to look at binding kinetics so that we, for example, when we are developing a biosensor and we look at our recognition element, that we know how this binding occurs and that we measure at the equilibrium. And also when we want to have a reversible sensor that can be able to sense continuously, then we have to have a closer or very close look to the dissociation rate constant, because this is going to tell you whether your sensor can be reversible in a reasonable time. So now the measurement principle of the technology that is called switch sense, we are an optical-based DNA biosensor. We have gold electrodes in which we place DNA nanolevers or DNA scaffolds that contain fluorophore for detection purposes. And in the other part of the DNA nanolever, we attach the ligand of interest. So when the ligand of interest interacts with the aldehyde, which is in solution under flow, then the chemical environment of the dye changes, and therefore we have a fluorescence change, which we correlate with the binding kinetics. Now with switch sense, we can also look at a series of other biophysical characteristics. So for example, conformational changes or enzymatic activity. But today I'm going to focus on the ones that have to be with FRET, such as ternary complex formation. First, I wanted to give you like an idea of how actually the machine looks like. So it's around this big, so benchtop. It has a series of user interfaces such as the autosampler, the buffer compartment, and the chip tray, which holds up to 5 chips. And everything actually happens inside the chip. So it's a microfluidic chip with a microfluidic channel on a single channel, and everything occurs in the middle, in the center of the chip that you can see there. And if you could zoom out, you could see the DNA nanolayers that consist in 3 different strands. So the anchor strand that always is attached to the surface, then the adapter strand that carries the dye, and the ligand strand that carries the ligand of interest. So with this setup, we can look at a series of biomolecules such as proteins, DNA, small molecules, etc. And now the typical automated workflow in the machine, it's the following. So we always start with the surface only with the anchor strand, and then we functionalize the surface with the ligand of interest. And this functionalization is very easy because it's just DNA-DNA hybridization. And then we inject a series of concentrations of the analyte of interest, followed by an injection of buffer to look at the dissociation. Then the surface is regenerated, and this step is also rather easy because we just inject a high pH solution that disrupts the DNA interactions, and we can start freshly with new ligand. Now I'm going to focus on 2 different applications of this technology. The first one is to look at secondary structures, and the second one is to look at ternary complex formation. So regarding the first one, I actually want to tell you a little bit more about the optics of the system. So we have 2 electrodes. The first one is the measurement electrode, and the second one is the control electrode that we use as a reference. And we actually have single photon counters for red and green channels in each of the spots so that we can measure up to 5 signals simultaneously. So this is why we can actually measure the FRET. Now, the previous talk already mentioned what is FRET, so I'm not going to go into it. But basically, the setup on the chip would be the following. So we have the ligand, in this case a nucleic acid. We have one dye, so the donor dye on one extreme, and the acceptor dye in the other side. And whenever there is a conformational change upon analyte binding, and this conformational change brings the dyes together, then we will observe a FRET signal. To develop this technology, actually we started with aptamers, and this is because aptamers are rather easy to handle, and many of you and many people use them as biorecognition elements, so actually they are quite interesting. And we chose this MN19 aptamer that was originally developed to bind cocaine, but yeah, we couldn't find cocaine, so we use quinine that actually binds the aptamer also. And the following plots are going to show you the actual data of the real-time conformational change of the aptamer. So as you can see, there is 2 plots, one in green and one in red, because we can measure the 2 signals simultaneously. So as the quinine binds the aptamer and the aptamer falls, we can follow the decrease of the green fluorescence and the increase of the red fluorescence. So whenever we have this increase of the red fluorescence, then we know that we have FRET, therefore the aptamer is folded. So we played around a lot with these type of setups. So we looked at, for example, different positions of the dye. We also look whether the dye was covalently attached to the aptamer, or if it was more kind of like hybridized to an extension of the aptamer. We also looked only exciting one dye, or exciting the other dye, or exciting only the green dye to get the FRET. And basically all this to say that the system was kind of versatile to different kind of setups that would suit different applications. One last thing that we did with this aptamer was actually looking at different small molecules that are very chemically similar to quinine, to see whether this aptamer was able to discriminate different molecules. And actually we saw very different kinetics, even though the small molecules were very similar. So this technique can also kind of give you kind of like a screening or a hit ranking, depending on the binding kinetics. Now the second application, this is a little bit more biologically relevant. So we tested a protein that is called IMP3 and is somehow related to cancer. So this protein is also interesting because it's a multi-domain protein. but we only look at the tandem domains KH1 and KH2, and this protein is known to loop RNA. So basically it binds to motifs that are separate in the RNA, and it bridges them together so that it creates loops. And this is one of the ways that is thought that the protein stabilizes mRNA in messenger ribonucleoprotein granules, so something that is generally not good. So in this case, the ligand, so this mRNA, was forming originally a secondary structure, therefore it was already in a threat-sensitive position. And in this case, then, when the protein came, what happened is that it actually disrupted this initial threat state by opening the loop and separating the dyes. So this is kind of like the opposite as what we saw in the previous example. The data look like this, so whenever we inject the protein, the loop is opened, therefore we recover the green signal and we decrease the red signal, so the threat is disrupted. And we also played around with the number of motifs, RNA motifs that the ligand had, because in nature actually it has several RNA motifs. So in this other example, we had 4 motifs for one of the KH domains, and we could observe the difference between the number of motifs, especially in the dissociation, and this tells us that this could be one of the reasons why the protein stabilizes RNA, because of a slower off rate and rebinding of the protein on the RNA. The second part of my talk is regarding ternary complex formation. And just as a little background, actually many types of drug modalities have been developed because there are many proteins that are very hard to target or to drug. One of them, it's called degraders. So these degraders are molecules that bind to one side to one ligase, for example, and the other side to a protein of interest, so that it starts the degradation of the protein of interest. And these, for example, are PROTACs, or proteolysis targeting chimeras, that is the name. So normally the assays that study this kind of proteins are similar to sandwich assays. So 2 of the components are pre-incubated and the third one is immobilized on a surface. This carries somehow some drawbacks, because whenever you look at the dissociation, especially, you could look at the dissociation of one of the partners or the second partner, or actually the complex. So you don't really know what is happening. So what we thought about was to actually immobilize 2 of the binding partners in a so-called Y structure that I'm going to explain in a moment, and then only the third partner being injected, so that we can only look at the kinetics of this third partner forming the ternary complex. So we do it with this so-called Y structure, and this Y structure actually has one arm with the protein of interest and a red or a green dye, and the second arm with the other dye and the ligase, for example. And it has also this flexible hinge region so that the arms can move and the proteins are not really restricted and they can move around and do their thing. So the assay looked like this. We always excite only in green, and whenever the PROTAC comes, or the degrader comes, and it engages the 2 proteins, then the dyes are going to come close together and we are going to see the threat signal. And actually, I really like to look at these plots because it's really intuitive to actually see one of the signals going up and the other one going down, so you can actually see the threat in real time. This is going to be one example of a very well-known system in PROTAC field. So the protein of interest is called BRD4, and the ligase is called VHL. The PROTAC is called AT1. So with this Y structure, we can also have a couple of different setups. For example, we can only look at the binary binding on the protein of interest. We can also look at the binary binding on the ligase. So we see here, for example, the kinetics are quite fast. And we can see then the ternary binding by looking at the threat. And of course, whenever there is this ternary binding and the PROTAC engages the 2 proteins, then we have this avidity effect and a decrease on the off rate that you see in this plot. Okay, so as summary, or what are the take-home messages of this presentation? First, that this kinetics FRET, or KIM-FRET as we like to call it, combines this FRET sensitivity with these biosensor advantages, which means continuous flow of analyte or molecule exchange. We have straightforward surface functionalization thanks to the DNA nanotechnology, and it can also work in a wide range of detection capabilities. So actually, the aptamer is in the high nanomolar range. The protein that I showed is in the low nanomolar range. So we can go quite wide. And it enables the real-time monitoring of the folding or binding-induced conformational change of different nucleic acids, so either aptamers or other structures. Second, the system is highly adaptable, so we can place dyes in different positions. We can also covalently attach the dyes or attach them in a hybridized manner so that they can suit different applications. And in this case, we showcase two systems, so protein RNA and small molecule DNA, so actually quite different systems, and it can be also potentially for other analytes. Then third, the FRET enables the analysis of ternary complex kinetics, and this is something actually that it's kind of new. So people has not been able to look at ternary complex formation before this Y structure. And finally, and I think in the context of this Congress about continuous monitoring, I think investigating on and off rates are very paramount to actually develop a specific sensor and to choose the right recognition element, because this, especially the off rate, for example, is very predictive whether your surface or your sensor is going to be able to regenerate or not in the time that you want it, and also it gives you a lot of information about the equilibration of the sensor. So this is something that should be normally done before doing your sensor to really characterize what is your biorecognition element and how the interaction occurs. So with that, I would like to thank you and be happy to take any question.
Thank you for that talk. Questions for the speaker?
Hi, thanks a lot for a very interesting and clear presentation. You described the cartridge, and you mentioned that there are two electrodes, one electrode and one reference electrode. And then in your talk, you showed fluorescence data, FRET data. Is it still useful to have an electrode? Do you use the electrode in any of these data, or do you not use the electrode?
Yeah, so let me go back to the setup, maybe. This one, for example. So we use the electrode because we apply negative potentials to have the DNA nanolevers in an upright position, so that they are more available to the molecules on the channel on the liquid. So that's one thing. And we have two electrodes. The second electrode is the reference, and for example, for this data is real-time reference. And we do this because actually the binding of the protein itself has an effect on the dyes. But we don't want to look at the binding, we only want to look at the folding kinetics of the nucleic acid. So we use the reference electrode to reference the binding signal of the protein. So the reference electrode doesn't have the red dye, it only has the green dye, so that we can reference that fluorescence of the binding, and we finally get these very nice mirroring plots, because if we wouldn't use the reference electrode, then the grid signal would look completely different, because it's a mixture between binding and FRET.
Okay, but the voltage is only used to align the probe, so is it a DC voltage that you apply to the electrode?
Yeah.
Okay. Yeah, thanks a lot.
But in other applications, we actually do the switching. That's
exactly.
Why
it's called the switch sense, right?
Yes, but in this application in particular, we just use constant negative voltage.
Okay, thanks a lot.
Thank you for a very nice presentation, very clear as well. I was just wondering about the dyes itself. So this is quite crucial to your technology? Do you have to sometimes screen for dyes, or do you have some dyes that you always use? How does it also influence your assays? So could you maybe tell a bit more about this?
Actually, you're right. They are like super important, and specific dyes will have a specific response depending on the binding partners. Some binding partners are gonna react, or some dyes are gonna react more to some interactions than others, and that's why we have a set of 6 different dyes, 3 dyes reds, 3 red dyes and 3 green dyes, and normally this covers most of the applications because the dyes that we choose have different chemical properties so that we more or less balance, and if one doesn't work, then we go to the next one.
Other questions? I will sneak in one last question. So you mentioned using these flat surfaces and how the binding does affect the dyes, right? So it's affecting your donor, so you basically have less donor fluorophore interaction for the fret to happen. So over time, do you lose sensitivity? Even though you have the reference, are you losing sensitivity if it's not just binding but fouling, such that if you're quenching your donor by some other means, do you lose sensitivity of the device over time?
Actually, the measurements are quite short. So for example, well, this one actually is like 1 hour, I think. No, it is not a problem because we can compensate for that, for example, by increasing the excitation light. Another way to compensate is, for example, regenerating the surface in each different concentration so that new ligands come and then they are basically, the surface is replenished. And basically these are the 2 things that can be done for that.
So are the measurements always taken in a clean sort of buffer, or are they actually taken in the sample matrix?
No, they are taken in clean buffer. So the samples are diluted in buffer, then uptaken, put into the microfluidic channel, and then in the dissociation, of course, is just buffer running. So this is an in vitro.
Gotcha. Cool, thank you. Any very quick question? Where I don't have to go upstairs. No? Okay. So let's thank our speaker, and then we'll move to our last speaker for the day.
Thank you.
I think we're right on time. While our next speaker gets set up, I'll do a brief introduction. So Andris Jeminejs is coming to us from Aarhus University in Denmark. He's a PhD student there. And he's continuing some of our talks now talking about synthetic RNAs and other nucleic acids for production of recognition elements and continuous sensing. He has many interests outside of, I think, research as well, as all good graduate students does, but interest in 3D printing, running, and a few other hobbies. So please get to know him. I guess hopefully he'll be here for later tonight and the other events. But without ado, Andris, thank you.
Thank you for the introduction.
Thank you for the introduction. And thanks for all of you for being here, both in person and people online. I know it has been a long day, so I'll try to keep it short. During the symposium, we have seen many examples of how both DNA and proteins can be used for continuous sensing. And especially during the last session, we have seen how combining both could yield some amazing results. So in this presentation, I would like to show you a method how we can actually attach the 2 very different biomolecules together to directly create continuous sensors. So in our project, we start with a protein. This protein is just, for example, an antibody or a part of the antibody that could bind the molecule that we would like to sense. Then what we do, we covalently attach DNA strand to it that ends with a competitive binder. And actually here you will see a lot of similarities with previous Tom Sau's presentation, a lot of parallels in our work actually. Afterwards, you can attach complementary strands that contain our optical readout. And here we have complete creative freedom. It could be a fluorophore and a quencher. It could be a thread pair. We decided to go with the thread pair for simplicity. Now, on its own, this assay exists in equilibrium where it's mainly in a closed state which results in a high FRET signal. However, when the analyte is detected, it displaces the competitive binder, which causes the whole system to open up, leading to the loss of the FRET signal. Ideally, we can immobilize the system with solid support or semi-permeable membrane, wash away the analyte, and the system goes back, and thus we have a continuous sensing device. And as our pilot study, we decided to start with a continuous sensor for drug digoxin that is widely used for treatment of heart disorders. And that is also a great example of a drug with a really narrow therapeutic range. So it makes a great target for these kind of sensors. As a protein, we decided to go with anti-digoxygenin full antibody or Fab. And Fab actually proved to be an even better version for simplicity reasons. And as a competitive binder, we found out that we could use the actual recognition unit of the drug, digoxygenin, to act as a bait, similarly how it is done in Tom So's work. So how can we make these kind of sensors? We start with a simple DNA strand, to which we then attach a competitive binder. And this could be done with easy NHS chemistry, which is a pretty standard method in this field. Afterwards, we need to attach some kind of reactive moiety to the other end of the DNA strand. And this reactive moiety will be used to create the covalent linkage to the protein. And in our work, we decided to target lysines on the surface of the protein. And for this case, as a reactive moiety, we decided to investigate either aldehydes for reductive amination, and we found some good candidates, as well as activated esters for amide coupling to react with the same lysines, and also found some good candidates. After creating this modified DNA strand, now it should be ready to attach to the protein. However, there is one step before that. We wanted to attach it regioselectively, and for that we need to control the geometry of the DNA strand. And we wanted to fold the DNA strand and lock its conformation so that the two ends are just spatially locked close to each other. So basically imagine like a clamp holding the DNA strand by its end. And we have investigated for this different tools and tricks of DNA nanotechnology to achieve this, and we were actually successful in this. When our DNA strand is folded, then we can mix it with a protein. And what happens first, the two come close together due to the competitive binder having some affinity to the binding site. What happens next, the reactive moiety now is basically pushed against the surface of the protein, creating high enough local concentration so that the reaction can occur only next to the binding site. So we have our covalent linkage and have created our DNA protein conjugate. And in this case, the reaction is exclusively affinity driven, which means that if we don't have a competitive binder or have some kind of other similar molecule, but that does not contain or that does not have a binding towards the binding site, the reaction just does not occur. And also, we observed that this labeling occurs specifically next to the binding site, and DNA prefers to attach to only one of the protein chains in the case of Fab or a full antibody. As well as the reaction proceeds with really high yields. The final step is really simple, just attaching the complementary strands with our optical readout, and our assay is ready. So when we took our assay and diluted it a lot and decided to take some fluorescent measurements, we found really nice FRET response. Then we proceeded with some titration and acquired these results. These look a bit wobbly, but if we take the integral values of the fluorophores and plot them, we actually see something that resembles an S-curve, which actually goes down to low nanomolar concentration, which is a pretty good start. We also wanted to investigate whether we can use the tricks and tools of DNA nanotechnology to try to investigate and maybe tune the assay a bit. So we looked at our assay and tried to play around, for example, with this region. So here we could have, for example, a single-stranded spacer that allows us a lot of freedom of movement next to the binding site. Or we can, for example, extend one of the strands to create double-stranded region, which is a bit more stiff. Or we can extend it even further, creating overhang, thus creating even more bulk. And again, all these small modifications are completely modular. They could be introduced with just a complementary strand. Let's color code them. And then if we look at the sensorgrams, how these different assays behave in the presence of the analyte, we see different behavior for each of these constructs, some of them having higher Fret values, some of them having steeper change, some of them having faster equilibration kinetics. So we can actually tune and trade different properties of the assay by changing seemingly really small things within the structure. Now that we know that the assay can respond to the presence of the analyte, and we can actually tune it, we decided to see whether we actually can use it as a truly continuous sensing device. So here we collaborated with a company, Medtronic, in Copenhagen, and we decided to play around with this kind of device where we have a channel where we could flow through our sample with the analyte, and inside this channel we seed capsules that are shown in pink, and inside those capsules that are made of semi-permeable membrane, we can trap our assay. And what happens, small molecules from the sample could pass through this membrane, go inside the capsule, interact with our assay, and pass on, while we can watch what happens in the assay with the fluorescence detector. And this is how this device or these channels look like. And there inside those channels we can see the capsules. And we actually made a small robot that could precisely navigate the fluorescence detector in front of a particular capsule to interrogate it continuously over time. So then, if we interrogate one of those capsules over a long period in time and see how it looks and how the Fret value coming from both fluorophores in the capsule changes, we could see that when we spike our sample with the analyte, we see a sudden drop in Fret, which over time recovers, and when we do it a second time after some period, we get response, although smaller, yet it is still visible and it still tries to recover. So we have a pretty strong indication that this kind of assay could be used as a continuous sensing device. And to sum up everything that has been done so far, so we have established a protocol how to label proteins with the DNA to create these kind of assays, and we can achieve those with high yields and high purity. These kind of assays have great Fret response, and by adjusting small things in the structure, we can actually adjust the properties and trade some of them. And we have investigated the potential for continuous sensing, which proved successful. And as a final thing, another advantage of this kind of system is that once we have assay for one molecule, it's rather easy to switch it to another one. All we need to do is to change the protein and change the competitive binder. And again, if we want to make another one, then we switch again. And right now, this is actually what we're doing. We're trying to expand the possibilities of this assay to try to target different types of proteins, because again, we do not rely on a particular structure of the protein. And also we're trying to cover as much possible analytes as possible, as much as possible. So yeah, with this, I would like to thank, first of all, my supervisor, Professor Kurt Gottwolf, for giving me the opportunity to work on this wonderful project. Then EU Horizon 2020 and Marie Curie Actions for Material Support, as well as Consens for really nice network. And finally, friends and colleagues back in Denmark in Group 106 for creating really nice work and life environment. Thank you.
Yes, it is. Hi, Andris. Thanks for your talk. You were showing the Fret results in the capsules. And these capsules, they are tiny, I guess, like a millimeter or something. It's a semi-permeable membrane, and the Fret probes are inside. And if you look at the kinetics of your data, that's a combination of transport kinetics and molecular binding kinetics. So which one do you think is dominating in the data that you have shown?
So what is known about this particular semi-permeable membrane is it takes approximately 2 minutes for small molecules to pass through. So actually the limiting factor is the interaction with the assay, and it is currently the challenge that we're trying to work on, because to be implemented in much more advanced devices, our assay is actually quite slow.
Mm-hmm. Okay, thanks.
Just to follow up on that, on the last bit of data, can you just comment on what the recovery was? I didn't fully understand, so the fret recovery.
So as you can see, it takes approximately as much time, or a bit maybe longer, to recover than for signal recover as it is to respond to the assay. And the kinetics here are not actually as simple as they seem, because there are actually two equilibriums happening in this assay. There is the interaction between the competitive binder and the protein, and once, for example, the competitive binder is out of the way, only then the small molecule could come in, and the same process but backwards happens during the recovery. So actually we need to kind of juggle and balance both equilibria in order to make the assay faster.
Gotcha. So there's two K-ons and K-offs.
Yes, exactly.
Got it.
And technically with DNA, we're able to play only with one of those equilibriums.
Yeah. Cool. Okay, thank you. Other questions? Straight away and steps.
Thank you very much for the presentation. Can you, do you have any indication if the binding on the lysine close to the binding pocket of the protein changes the KD of the protein? Does it impact actually the affinity of the protein?
Yes, because DNA obviously creates steric bulk. And also, we are not still sure about the true geometry of what is actually happening, because a lot of literature tries to draw these kind of conjugates, like, if I can get the slides.
I'll believe you.
Yeah, so a lot of literature, it was, they try to draw it as if the DNA facing outwards of the protein, when in reality, actually, DNA is negatively charged, while these kind of large proteins like antibodies are usually positively charged. So you can imagine them wrapping around the protein in a way that is very unpredictable. And obviously, that could affect the binding of the protein.
But have you compared, for example, like the apparent KD with your DNA label and without?
Yes, so we have done some BLI experiments, and having DNA there is the slowing, the limiting process, so it is slower.
But only on the K off, but not on the KD, like the thermodynamic constant.
So if we're talking about the interaction with the analyte, first happens the KD off of the competitive binder, and then K on of the small molecule. So that issue we can solve by creating more bulk, which makes it faster to dissociate for the competitive binder. But when we are talking about the recovery, we need to remove the small molecule, which is something that we have very little control of. That is something only that we can control with media, with salt concentration, to basically wash out the small molecule. And then the competitive binder from the DNA comes in with KD on. So actually, yeah, depends which process are you talking about.
Of course, thanks.
Other questions? Was there anything online? No? Okay. If there's no other questions, I think we'll turn it over to our host for some closing remarks. And let's thank our speaker one last time.
Okay, guys, we are about to close the first international symposium on continuous real-time biomolecular sensing. And since it's the first symposium, we should try to learn from it. And I think, what is very clear, what unites us is this dream of trying to realize sensors that, in the future can measure other substances than only glucose and pH continuously, like, the many small molecules, the pharmaceuticals, the environmental pollutants, the inflammatory biomarkers, proteins. And I'm very happy that we've seen very recent results and early peeks into future potential applications, and also that many people have attended. So the audience is now not that big anymore, but when we started yesterday, it was nearly a full hall. And the live stream of yesterday was viewed in cumulatively 600 times. So it shows that there's really an interest in this field. So yeah, that's very good to see. In terms of the topics, we decided to focus on the core of the technology, which means the molecular concepts and the detection concepts. And well, we've seen a wide variety of those: electrochemical, fluorescence-based, particle-based, plasmonic, Raman. And we've seen that most sensors are based on switching principles, and there's lots of engineering challenges and intricacies with these switches. And we've seen very often the trade-off between sensitivity and speed, so affinity and reversibility. And we've seen interesting reversibility approaches. Of course, there's the tuning of aptamers and of antibodies, but then there's also the use of elution buffers, of electric field and electric current, and using temperature. So it will be interesting to see how all these different approaches, how they develop in the coming times, and yeah, what will serve us as good solutions. Sensitivities, we've seen micromolar, we've seen nanomolar, we've seen picomolar, so it's really all over the place that people are addressing these challenges. Response times, seconds, minutes, tens of minutes, and longevity, so length of operation, we've seen hours, we've seen days, and I'm sure that next time we'll also see weeks, because yeah, that's a matter of further technological improvements. In terms of opportunities, many talks were justified or introduced via medical applications, patient monitoring, but also medical research, and then also industrial process monitoring, biotechnology, food industry. And all of these are demanding in two aspects. It's the integration. So how can you make a device that really is integratable with this application? And for example, with patient monitoring, miniaturization will be very challenging. In an industry, you have other device-like challenges. In all cases, the analytical performance will be very demanding. You have to measure in the right range, with the right precision, with the right accuracy, with the right calibration strategies. All of that is really, really a lot of work. Most mature is the EAB technology, the electrochemical aptamer-based, for small molecule monitoring. It's now moving towards pharmacological studies. It's interesting to realize that this was conceived 20 years ago. So it's taken some time. And I think Tom So said that the electrochemical glucose sensor took 40 years, or yeah, a very, very long time. Well, let's say 40 years, and then 20 years, and then maybe present-day technologies will take 10 years, or even somewhat less, because yeah, we can expect that those developments will go faster and faster. Marien de Jonge yesterday gave us the medical perspective, and he talked about the chicken and egg problem. If a doctor doesn't know that something is there, or doesn't know how to interpret the data, the doctor's not going to use it. So yeah, we have to really work on the technology, to validate the technology, then to validate the clinical application, and then get to the clinical acceptance. And those are really, yeah, that requires a lot of work and long time scales. So in fact, there's many reasons for us as sensor community to work together, to learn from each other, share ideas, share experiences on science, technology, and the applications. And in terms of applications, I think we are lucky, because there's tons of applications. The application space is just very, very large. So there's room for all of us for applying the different technologies that we are all developing. So how to proceed? We would be very happy seeing how the conference has gone, the attention, what people have shared, the discussions that we had, the lively discussions. We will be willing to organize this symposium again next year in the framework again of consens and sense us. But first we want to listen to you, so you will receive a feedback form by email, and please give us your frank opinions, your wishes, your remarks, and we will take that into account for the next edition. On a side note, we'll organize a one-day summer school at the upcoming World Biosensors Conference in Lisbon, May next year, focusing again on continuous monitoring of low concentration substances. And on another side note, scientific journals are also picking up on this topic. For example, the Biosensors Bioelectronics has a special issue now that focuses on continuous monitoring, but that's both physical sensors for continuous monitoring and chemical sensors. So that's, in fact, much broader. So it's time to close, and I would like to ask you to join me in thanking many people who have contributed to realizing this conference. So with intense appreciation, the consents team, the Sensors team, the speakers, the session chairs, Eindhoven University for its support, people online, and of course the team for audio and video. Thanks, Mark and team. Let's give them a big applause. So tomorrow, many of us will be back because we have the final contest day, final event of the Sensors competition. That's going to be very exciting, with pitches from the student teams about technologies and applications. But other people will not come back, I guess. So I close the symposium. I wish you a good reminder of the day for people online. For us, the evening, and maybe for others, maybe a continuation of the night. I hope to see you soon at another occasion in this fascinating field of continuous biomolecular sensing. Thank you very much.